software. Lifetime, it was a Nature review which was shared. And the diabetic neuropathy has quite particular symptoms that you can see on screens. I think it has been shared. So the tingling sensation, as you can see on the screens.
Dr. Surabhi, sorry to disturb you.
Yeah.
Slides are not moving.
Slides are, so okay. So let me escape this.
It's moving. It's okay now.
Okay. Okay, fine. So there are incidences of neuropathy in diabetic patients that can range from 10.5% to even 32.2% as per the data as reported in India. And these symptoms, they include the sensory numbness, and the pain cannot be felt sometimes, and the person is not even aware sometimes that he or she is suffering from diabetes. So tingling sensations, pricks and needle kind of sensations, burning sensations, and then sometimes no sensations that results to the numbness and increased sensitivity, slow healing wounds. These are quite complicated in diabetic neuropathy. So when we talk about the symptoms, we also come across that certain, so I will share the next slide. So the diabetic neuropathy has developed.
Dr. Surabhi, can you please make it full screen?
So when we are making the full screen, the slides are not changing.
Okay, no issue. You carry on.
So I think it's visible. I've done 87% of resolution. So I think it must be available on the screens clearly. So the development of diabetic neuropathy takes place through a very weird kind of pathways, which result into the reduction in the DRG, which are the dorsal root ganglions. So ischemia and other complications which are resulted due to hyperglycemia actually result into the complications in the sensory neurons, which we can see on the screens, and particularly the DRG region, and also the motor neurons. But of course, the sensory neurons are more affected, and sometimes even it causes the demyelination in the neurons, and sometimes even the Schwann cells may be affected, and it may lead to the degeneration of the neurons. So the reduced kind of Schwann cells axonal transport is also a remarkable feature of this diabetic neuropathy. This is again a figure which was shared by the Nature reviews itself. And the next, we can see that what is the other complications and what is the diagnosis of this diabetic neuropathy. So the we can test out the diabetic neuropathy with the help of the nerve conduction velocity test. If there is a deficit, of course, there is a diabetic neuropathy. And if there is a demyelination, as I have shown in the previous figure, then also that can result into the decreased myelinated fiber or intraepidermal nerve fiber densities. That may be tested as a diagnosis of DN, diabetic neuropathy in short. So there can be behavioral changes, and that we can assess in case of the mice, and some anatomical features can be also changed during the diabetic neuropathy. So the sensory functions that are determined by certain simple kinds of behavioral tests like cold allodynia test, the hyperalgesia test, the hypoalgesia test, and various other tests which we have conducted in our laboratory can confirm the pathogenesis of diabetic neuropathy. So through the behavioral responses itself, we can determine that whether the diabetic neuropathy has been caused or not. So let's look at the mechanisms underlying the diabetic neuropathy. So why does this diabetic neuropathy occur and how does it proceed? So hyperglycemia, increased blood glucose, obviously greater than 200 mg blood per deciliters. That much of the high glucose can lead to the protein oxidation and sometimes even the lipid oxidation. So the hyperglycemia resulted due to the high glucose may sometimes result into the abnormal ROS generation and the reactive nitrogen and reactive oxygen species generation that may lead to the DNA damage, even the endoplasmic reticulum stress, even the mitochondrial dysfunction, which we are going to deal in with in our study, and also the cellular injuries and the irreversible damages to the neuronal cells when we talk about the neurons and the brain system. Also, not only this, but dyslipidemia leads to lipid oxidation, and this is followed up by increased LDL, decreased HDL, which are high density lipopolysaccharides, increase of the free fatty acids, and that may altogether result into a pathway which leads to the increase in the inflammation at the sides of the neurons. And also, not only this, there is some effect on the insulin signaling mechanism via the effect on PI3K and AKT pathway. So these all mechanisms actually lead to the stress on the neuronal morphology as well as neuronal functioning, and sometimes even neuronal apoptosis that may lead to the death of the neuronal cells. So what are the traditional therapies, existing traditional therapies, and are they sufficient for controlling the disease? So as we have seen till now, this is again data which was shared from the Nature paper. The traditional therapies that are existing are anticonvulsants, some SNRIs, and the tricyclic antidepressants. But they have only the tricyclic antidepressants, they have partial effect. Anticonvulsants, they almost have no effect. And when we look upon that, what are the US approved drugs? So we come across only duloxetine and pregabalin, which are used for the traditional therapies in the diabetic neuropathy. So there was a loop that can we have some natural therapies which do not have any side effect or cannot cause the injuries to the other cells nearby. So we come across certain kinds of natural therapies also, which are adopted as the natural alkaloids. They can be used in the therapy, and cocaine, morphine, piperine, serotonin, and caffeine to some limited extents can cause decrease in the pathogenesis of the diabetic neuropathy. Then also zeaxanthin and astaxanthin and lutein. These are the antioxidant carotenoids, particularly the xanthophyll carotenoids, which can protect the eyes from inflammation and oxidative stress in the retina. And out of this, we have chosen astaxanthin. But why astaxanthin? Why not zeaxanthin? Why not lutein? So we have chosen astaxanthin because it is a unique member of xanthophyll family, and it is, as you can see in the structure, it has got two rings. So it has got a symmetric molecule structure with two terminal rings that are joined with a short polyene ring. The hydroxyl group are not only one, just two of the hydroxyl group that are present on the structure of the molecule on the rings that enables it to esterify the fatty acids to form the monoesters and diesters. Now, this is a remarkable feature which is having an advantage over other xanthophylls. Other xanthophylls are having only one OH group. So, astaxanthin is a unique member which has got two OH groups. Now, what is the advantage of this group? So this helps in quenching this hydroxyl group is accepting the electrons, and then it is stabilizing itself. This whole structure is stabilizing itself. Now when it does so, it is actually converting those ROS molecule into the non-toxic ones, like the singlet oxygen into the non-toxic ones. So in due course, this is a very helpful molecule for controlling the oxidative stress and detoxification within the neuronal area. So the neuronal morphology is also protected, and these experiments are ongoing in our laboratory. We are also seeing the morphology of the neurons. In the next probably lecture, we will be able to share this data, not in this one. But of course, we have also got some lead about that also. So astaxanthin is a unique member of xanthophylls, and it has got deep red color, phytonutrient, which is usually synthesized by the microalga known as Hematococcus fulvialis, and also it is existing in the microalgae, complex plants, and also the seafood, shrimps, etc. So that is the reason why we have chosen astaxanthin. So with this clarity, we also looked about what is the mechanism why astaxanthin works so beautifully in consumption of these ROS. So actually the acute brain injury or the neurodegeneration in many of the disease is due to the ROS generation within the milieu of the cells of neurons in the brain. And therefore, this astaxanthin actually, it activates the PI3K pathway, and via AKT, it is going to activate NRF2. And this NRF2 actually does the transcriptional activation of the pathway, ARE pathway, and it leads to the mRNA production, which leads to the synthesis of certain very important proteins like GST, alpha-1, etc. And this leads to an inhibition of the oxidative stress in the milieu of different kinds of cells. So particularly, it has been seen in the case of the other neurodegenerative disorders. So that is why we have chosen astaxanthin. Not only this, but it also inhibits the SP1, which is working as a promoter for the harmful proteins that can cause the diabetic neuropathy. So action mechanism of astaxanthin, when we talk about it, actually inhibits the ROS, and the ROS is involved in the mitochondrial dysfunction very importantly, which we are going to talk about in this particular study. So mitochondrial dysfunction is associated with the ROS generation, and it is a remarkable feature of the diabetic neuropathy pathogenesis. ROS often leads to the inflammatory signaling, leads to the cellular dysfunction and cell death, and sometimes even the mitochondrial apoptosis, which leads to the disease development and progression. And of course, this may result into several kinds of inflammatory diseases, several kinds of cardiovascular diseases, neurodegenerative disorders, not only limiting to diabetic neuropathy, but other ones also, and liver diseases and other metabolic complications, and of course, aging. So when we started this study, we had obviously very clear objectives that we have to study the effect of astaxanthin on behavioral changes during the diabetic neuropathy pathogenesis in the STZ-induced diabetic mice model. Then we had to assess the oxidative stress in the brain sections of the STZ diabetic mice, and we had to assess the effect of AX, astaxanthin, on the control of this oxidative stress, and we had to determine the effect of astaxanthin on mitochondrial dysfunction, because mitochondrial dysfunction, as we have already seen, is taking place in the neurodegenerative disorders in the different brain sections. So we have taken different brain sections like the cortex, the cerebrum, the cerebellum, and the hippocampal lobes for the assessment of mitochondrial dysfunction also. So now let's look at the material methods in very short. So animals were taken, the adult male Swiss albino mice were obtained from the University of Hisar and Veterinary University, and then diabetes was induced in these mice, and with the help of the STZ injections, 40 mg per kg of the STZ was given consecutive for 5 days for the induction of diabetes, and then later, with the help of the GOD-POD kit, the plasma glucose levels were estimated in these mice for the confirmation of the diabetes. So the mice which were having greater than 200 mg per deciliters of the level of plasma glucose, they were considered as diabetic, whereas other ones were given some further injections to confirm them as diabetic. So then the mice were not just right away used, they were left for some 2 weeks for the development of diabetic neuropathy, because that doesn't happen in just one day. And thereafter, what we have done next is we have divided the mice groups into the diabetic and non-diabetic mice. And of course, thereafter, we have done certain behavioral assessment studies on these mice for the confirmation of diabetic neuropathy. So the mice were divided into the DD control and the non-diabetic controls, and then the different groups of diabetic mice were induced with were administered with the astaxanthin in the increasing drug doses of 2 mg per kg and then 4 mg per kg for just 4 weeks. And this whole experimental protocol was approved by the institutional committee also. Then thereafter, our next task, and that was a bigger task, that how to confirm this, that diabetic neuropathy, yes, has been developed in case of the mice, the diabetic mice. So we confirmed this with the help of certain behavioral tests. So tail immersion test, the hot water and cold water testing was done. So for hot water testing, we have taken 55 degrees Celsius water, and cold water nearly 4 degrees to 10 degrees water was taken for this tail immersion test. And then cold allodynia test, that was on a tail flickering test on the cold plate was used. Then Eddy's hot plate test and the Randall Salito test were performed. So this is just a small view of how the tail flick test was done. Next, our target was to assess the oxidative stress in terms of the different antioxidant enzyme levels, and this was done by determination of the superoxide dismutase, catalase, glutathione peroxidase, GPX, and the reactive oxygen species. So not only this, but we also looked for the nitric oxide production, lipid peroxidation, higher redox and reductase activity, and also the acetylcholine esterase activity in the different brain sections. Also, the different brain sections that were assessed from the mice after their ethical sacrifice. So after the sacrifice of the animals, the brain sections were also utilized for the mitochondrial functional assays that were done, which are as follows, and then we come to the results of this study. So as we can see, our first target was to just find out a very big thing, that is a body weight. Was there a change in the body weight? Yes, there was a change in the body weight, and the diabetic control was having a lesser body weight as compared to the other normal ones. Right? And when we administered the astaxanthin, there was a slight improvement, not slight, but a significant improvement in the body weight. And thereafter, we had also assessed the blood glucose levels. So we have found that, of course, in diabetic mice, the blood glucose level was significantly lowered by the administration of this astaxanthin, and this may be the cause why this astaxanthin has worked out in our case. So thermal sensitivity was tested in mice with the help of the tail immersion test. Like in hot water test, we had conducted this test in our laboratory, and as you can see, the mice paws are just abruptly it has shown a reactiveness towards the hot water test. Then it was given the astaxanthin. So as you can see, the left mice is representing the diabetic control mice, and the right one is representing the diabetic mice, which was administered with high dose of this astaxanthin. So you can see abrupt sensory mechanism has come back to this mice, and tail immersion test was successful. Then we can also see this test in a short movie. It can run. I think it cannot be shared. Let me just see whether it can be shared. So we have also conducted this test in our laboratory using a video clip. But I think that is not playing. I'll show you this in another format when we see. So this was the test which was conducted. Then effect of AX on tail immersion test. What was the result? So when hot water was used for the tail immersion test, again the same results were found, and hyperalgesia test, when it was done, it was found that diabetic control mice was having a much lesser latency period as compared to the non-diabetic control. But it was normalized when we gave the increasing doses of the astaxanthin. The next, we have tested this cold sensitivity test, again by tail flickering test. So as you can see, the upper ones are diabetic control mice, which shows almost no tail flicking during the cold sensitivity. So what we can infer from this, we can infer that cold sensitivity is lost in the diabetic mice. The sensation is lost. Obviously, that may be the play of DRG of the dorsal root ganglion of the spinal cord, that they might have lost the sensitivity. And then you can see this has been improved by the administration of this astaxanthin. So you can see slight flicking in the lower mice, which was administered with 4 mg per kg of astaxanthin for near about 4 weeks. So maybe it could have even more increased when we had looked for 6 weeks, but most of the studies have been limited to the 4 weeks. So that is why we have chosen this time period. More of it was the effect of AX on cold allodynia test. We have seen the results, so we are not going to discuss it again. Now, the diabetic mice, that was also showing a very good response in the Eddy's hot plate test after the AX administration. So can you please just guide me? This video is not running. So is there any way to place this video? This is a video clip which I have included for this Eddy's hot plate test. Is there any way that I can play this? If no, I'm just going to summarize it, that Eddy's hot plate test is done in case of mice, and we can see a slight picture that the mice tail is flicking under this Eddy's hot plate test. And this explains that the sensation is regained after the astaxanthin administration to the diabetic mice. So as you can see in the graph, the astaxanthin administration in the increasing doses has resulted in the betterment in the sensation in terms of the tail being flicking, regained its functioning to the sensory neurons when it was tested upon, when the mice was tested upon the Eddy's hot plate test. So that was the Eddy's hot plate test. Then the Randall Selito test. Again, the same kind of results were found, and we have seen that significant increase in the sensation was brought about in the astaxanthin induced diabetic mice. So when administration of this astaxanthin is done, also the antioxidant activity has been increased. Now this was confirmed by taking the different brain samples from the different mice, groups of mice, the diabetic versus the non-diabetic mice. So the diabetic neuropathic mice were sacrificed, and then non-diabetic neuropathic mice were also sacrificed, and then the different brain sections were taken out, like the cortex, cerebellum, striatum, and the hippocampal lobes, and then they were studied for the effect of astaxanthin. So astaxanthin was already administered for 4 weeks, and we had taken out these samples. So then the brain sections were homogenized, the tissue was homogenized, and then the supernatant was taken for the estimation of different kinds of antioxidant enzyme as a mark of the biochemical estimations and enzymatic estimations for the measurement of oxidative stress. So the SOD activity was found to be increased after the astaxanthin administration in the increasing doses of 2 and 4 mg per kg of the doses, and we have seen significant increase, as you can see in this graph. The next astaxanthin activity on GPX was having almost a similar result as that of the SOD, and particularly the good results were found in all the sections, not only cortex, cerebellum, striatum, also the hippocampal lobes. So as you can see, this was also increased. The GSH activity, the glutathione reductase activities were also increased, and also the catalase activity were significantly increased. However, we have found much better results in case of the hippocampal lobes in case of the catalase activity. While this was so, we have to still look for that why not other sections of the brain had responded in the same way. Now, the another effect was on reactive oxygen species, and we have seen that even better results somehow were given by the diabetic mice, and we have seen that significant increase in case of the diabetic mice. There was a significant increase in the oxidative stress control. Then also the lipid peroxidation. Lipid peroxidation was decreased, but not to the level of the diabetic, non-diabetic mice. This was something which was weird, but since more of the results were focusing upon that catalase was, so not all the results are very close to what you want. So LPO was something which was different. Then NO level was little bit increased. This also we could not explain on what we got for the other results. But of course, nitric oxide has a dual role. So it plays both a positive role as well as a negative role. So in this study, we have taken the positive roles of NO, nitric oxide, and they might have increased. So administration of astaxanthin has increased significantly the NO levels, nitric oxide levels, as a biochemical molecule. And then effect of astaxanthin on acetylcholine esterase activity. Now this confirms the functioning of the neurons, and of course, as you know, acetylcholine esterase are the enzymes. So they are the acetylcholine esterase is also increased significantly in case of the diabetic mice which were administered with astaxanthin. Not only this, but also as a mark of the mitochondrial activity also, we have found that TRR levels are increased, and then MPT reduction assays. Now we come to the mitochondrial dysfunction. Mitochondrial dysfunction is of course related with the neuronal damage and sometimes even neuronal apoptosis. So if the mitochondria is not functioning, the cell will not work, right? So the mitochondrial dysfunction was estimated indirectly with the help of the MPT reduction assay, and it was found to be, as this result has shown, that increase in the activity of MPT was there. And then cytochrome C reductase activity. Now we know that different Krebs cycle enzymes are involved if the mitochondrial activity has to be checked. So mitochondrial activity can be checked by indirectly estimating the mitochondrial enzymes, the Krebs cycle enzymes, right? So the NADH cytochrome C reductase complexes number 1 to 3 activity can be administered, and therefore we can confirm, okay, the mitochondria functions good or not. So the effect of astaxanthin was good, and it was significantly increasing the cytochrome C reductase activity in the different lobes of the brain sections. Then effect on SDH was also increased significantly. So we have seen that 4 weeks of treatment with AX has increased the activity of the succinate dehydrogenase activity, and also the citrate synthase activity was significantly increased after the administration of this astaxanthin in the diabetic mice. So with this all data, we could conclude that in the present study, we could evidence that there are modulatory potentials of this astaxanthin, of course, the natural compound, which do not have any side effect. And the plus point is that it does not have any side effects on the other cells. And it also controls the oxidative stress and improves the mitochondrial activity by enhancing the mitochondrial enzymatic activity in the various brain sections of the diabetic mice. And he suggests that, of course, astaxanthin can have neuroprotective properties. Of course, that can be tested further. We can also test that whether a pre-administration of astaxanthin before the disease could develop fully can also contribute to this particular amelioration before it happens, and that can be largely attributing towards its anti-inflammatory and antioxidant properties, which can be looked further in the case of the diabetic neuropathy. So with this, I conclude this talk, and of course, these are the references, and this work has been communicated to one of the paper, and it has been accepted. I'm glad to share you. This was not the complete data, but a part of that. So the next paper has got, it was just a lead, and we are working now upon what kind of genes are involved in the in regulating these astaxanthin functions, and how those genes can cause an amelioration and whether not for the diabetic neuropathy. So with this, I come to an end, and thank you so much for being with me and sharing my ideas on diabetic neuropathy and its control by astaxanthin. So thank you so much. And this last picture shows my department, of which I'm a part, Department of Biosciences and Biotechnology, Banasthali University. Thank you so much. And special
Thank you,
thanks
Doctor.
to Dr. Rajkumar, who has given me a privilege to be a part of this international platform. And I thank you again, all the members of the committee we have, which have been integral part of organization committee and have done work so good for compilation of such a good level of talks. Thank you so much.
Thank you, Dr. Surabhi, for your nice talk. Uh, as chairperson, I have basic query because I'm a scientist and astaxanthin we know about is extracted from algae. Uh, Particularly, you mentioned that legal name. So my query is like this astaxanthin is you studied in diabetic mice. It is linked with kidney and other organs of the body. Means it is beneficial to take astaxanthin or what are the dose of astaxanthin?
Very good question, sir. Actually, there are 2 points. So this question covers first point. Of course, astaxanthin can be also helpful for the control of other oxidative stress in other parts also, as we have seen. In retinal cells, it has shown a remarkable activity. Like in case of the retinal cells, it has almost cured the retinal cells from the diabetic conditions. So diabetic retinopathy when we talk about. So of course, astaxanthin can work out for other organs. Number 2 point is that astaxanthin has got a little 1 point which is negative, that it is sparingly soluble in water, more soluble in DMSO. So we have given the doses in DMSO and not water. And the limit of dose was below its LD50, of course.
What
I have a question.
other antioxidant to patient
Uh, can you please repeat
if we
again,
don't
sir?
have
It's
astaxanthin?
echoing. It's echoing, sir. Can you please write your question in the chat box if it's
Now
possible?
session is open for other question. If there is no question, we can start next.
Dr. Surbi, I have a query. Like you check the nitric oxide levels. So was it at the molecular level or was it by the enzyme nitric oxide synthase?
Yeah. So it was actually the grease method, which was used in the tissue supernatant. As I have already talked, that most of the biochemical assays were done in the tissue supernatant. So the various brain sections were taken and it was homogenized, then centrifuged for 5,000 RPMs and so for 10 minutes. And then
Got
the supernatant was taken and then supernatant was analyzed for this NO level with the help of the grease reagent.
Okay.
Yeah.
I have one request also, ma'am. You showed me you showed some experiments which were not like video was not playing. So can
Ha.
if it is possible, you share the screen and you
Why
can
not?
play the
Why
videos.
not? I would be
That will
delighted
be
to.
very nice.
I would be delighted to if I can. So I have got files of those
So
videos.
you play the raw files and share the screen. You
Okay,
can.
okay, fine. Why not? Why not?
That will be good.
I'm not sure whether I will be able to, but I can try. Of course, I can try. So there was a tail flicking test. Let me just rule out whether actually I have got too many videos and I am just not very sure which I can show. Okay, yes, this can be showed. There is a plethora of videos for the behavioral test, which were, of course, conducted in my laboratory. So I think this is quite visible now. So this was the Eddy's hot plate test which was conducted. So this was hot plate, and the mice was so that the mice is not so insecure. So we have covered the face of the mice, and we have just tested upon the tail by touching this to the Eddy's hot plate just for a few seconds, which was ethically approved. So as you can see, the mice after the astaxanthin administration has shown the flicking, as you can see. So can you see the flick in the mice tail? And this was the beautiful result we got in this particular experiment, that after the astaxanthin administration, the flicking in the tail of the mice was much more faster, and the sensitization has been really caused. So this was a very good result which we got. We have
Thank you.
other videos, but I think
Yeah,
the
yes.
speaker has to wait them a lot. So I
Thank
think
you.
it was visible to you.
Yeah.
Thank you so much.
Dr. Surabhi, did you check the biochemical parameters in some other tissues also, other than brain?
True. Sir, actually, there was a suggestion from my PhD scholar itself that we can also test it, that whether astaxanthin has got some effect on liver. So we got a certain lead that it has got effect on the liver cells as well. But that data is just a lead. So we are working, another project is about this lead that astaxanthin can
Yeah,
have.
whether it is having any toxic effect also on serum parameters, liver function test, renal function test, etc.
So for this, there was a preliminary data, and we just tested out from an external source. Since that time we did not had the cell culture facility in our, but now it has been developed in our university, in my department itself, and we are pleased that there was a to share that there was no such toxic activity shown by the astaxanthin in the cells, which were the Kupffer cells when we talk about.
Okay, thank you.
Thank you so much. And special thanks to Dr. Rajkumar and the HOD of department for inviting me for this seminar and sharing my thoughts on diabetic neuropathy. So I was so delighted to be with you people and share my thoughts on this topic.
Thank you. We thank you too. Thank you.
My pleasure.
Thank you, Dr. Surabhi. Now I welcome our next speaker, Professor Oliver. Very good afternoon, Professor Oliver from India. So I'm giving very brief introduction to introduce Professor Oliver, all the participants. So Professor Oliver is integrative behavioral neuroscientist who has been working on animal models of affective disorders with a focus on emotional dysregulation, learning and memory with behavioral and autonomic readouts. He has high interest in translational animal models in biomedical research with technological development beyond mainstream. As animal welfare officer, he also supported animal welfare relevant improvements, particularly on the basis of his expertise in behavioral neuroscience. He obtained his PhD in 1991 on insect behavior at the University of Marburg, Germany. He was Humboldt Fellow in Slovenia and University MIUSC during 1991 to 1993. He was senior research scientist at the Max Planck Institute for Experimental Medicine, Göttingen during 1994 to 2004. He was research group leader since 2001. He was Werner Grant guest professor at the Karolinska Institute, Stockholm, Sweden, 2003 to 2004. He served as an assistant professor at VU University Amsterdam during 2004 to 2005. He was associate professor and head of the behavioral and cognitive neuroscience research group at the VU University Amsterdam during 2005 to 2016. Since 2016, he was the chairman of the animal welfare body VU and VU Medical Center. His expertise is aversive learning paradigm, fear conditioning, passive avoidance, fear circuitry, autonomic function, serotonergic modulation, neurocardiology. He is a member of Neurofederative Dutch Deutsche Neuro Federation, if I am wrong, please correct. European College of Neuropharmacology and NWG. He has many papers in reputed journal of neuroscience like Neuroscience and Behavioral Reviews, Behavioral Brain Research, Nature Neuroscience, Brain Research, Journal of Neuroscience, Front-End Pharmacology, Neuroscience, Letters, Stress, Brain and Behavior, and many more, and has more than 4,000 citations to his credit. So now I welcome Professor Oliver, and it's a great pleasure for all the participants from India to hear the talk of Professor Oliver title. And Professor Janakahi, if you want to say something, you can add in that.
Oh, thank you very much for thank you very much for the invitation. Maybe Janak still has something to add.
Yes, we welcome you, and we are very much looking forward to listen to you, Oliver. It's nice to see you again. That's great. Please welcome.
So thank you very much for this kind invitation, and don't exaggerate. I'm a humble scientist, and I'm always open to be addressed. And if in any case of any questions, please connect or check with me. I have my email provided, and I will share now my screen. I hope the sound is fine and that the communication is good, and then I will share a couple of aspects from my presentation now. Now let me check that I can share my presentation with you. Here it comes. Can you see this?
It's coming. Yeah, now
coming.
it's fine.
Great. So I will split up my lecture in essentially two parts. I will give you some ideas in my first part on the behavioral background, where I will give you some ideas about developments.
Sir, sorry to disturb you, sir. Will you please make it full screen?
Yes. I have. This is now full screen. Does it work?
Not yet.
Is it full screen now? Can you see this?
Sir, not yet. Sir, you can once you can stop sharing, and then you can give a retry. Again,
Okay,
you can reshare it. That will, I think, solve the problem.
Yeah. Okay. Let me check.
Sometimes due to connectivity issues, such type of problems arises.
Okay. Then I will say full screen share. Let's see. And then let me check. Is it working now better?
Now it's fine, sir. It's full
fine.
screen now. Okay, thank you, sir.
So, so I will give you some idea about some classical tests to assess anxiety, fear, and memory. I will give you some ideas about pitfalls of commonly used behavior tests based also on my experience. I will give you an important viewpoint on the interaction between cognition and emotion, because if you want to assess emotion and let's say emotional disorders that also affect on emotion, they will interact with cognition to some degree. And then I will give you an idea how we can better separate this by removing many unspecific stressors based on home cage-based phenotyping. And I'll give you some perspective, because there is a big initiative in Europe and also now starting in the USA, where we are in the move of more automated phenotyping in the long run. Just to give you a basic principle, of course, we talk about from neutral to aversive and pathological emotional states, in that we characterize, of course, a relaxed state, an attentive state. We refer to increasing arousal intensity, where vigilance is increased with increased attention alertness. Then we talk about anxiety, that is triggered by a potential threat, or fear, which is triggered by an explicit threat. The dilemma comes from all the pathology terminologies coming from the clinics, because whether it's pathological anxiety or panic, all these are characterized based on different durations as emotional pathologies in anxiety disorders in medicine. And of course, we have to be aware that if there is a panic to let's say a panic response to or spider phobia, that is explicit triggered by an explicit threat and should be different from something that is let's say, intrinsically, where we have cognitive appraisal without an explicit threat in an ambiguous-like situation. And of course, as you all know, that these centers and changes in the underlying neurotransmission, particularly the serotonergic system, are implicated in the etiology of mood disorders, ranging from a broad spectrum. And I'll give you some ideas why we think certain assays that we are using are really well suited and some are not so well suited, particularly for the translational aspect. And I just remind you, the classical anxiety tests that we use are, for example, the elevated plasmase, where you put an animal on the center area and you ask it to explore, and depending on the exploration pattern profile, you will argue whether it is a more anxiolytic or an anxiogenic-like behavior, anxious-like behavior. The same strategy is often applied in the zero maze, where you have areas which are protected versus unprotected areas, and as usual, you would always expect that animals that are not anxious explore the open areas. Another classically used test is the open field test, where animals are placed in an arena. Ideally, it's a round arena, not a square form arena, and of course there is an illumination, and we again look whether the animals show high levels of thigmotaxis, which always is an indication that there is an increased level of anxiety. And last, there is another system which is called the dark light or light dark box. I will show you later that many papers have been published on the so-called light dark box, indicating that the animals are, let's say, placed in the light box and then they are able to enter the dark compartment. And of course, I will show you data where I recommend, also based on other studies, if you use this test, start out from the dark compartment, because this allows you a better approach with higher validity, as I will show you in some results. Of course, the problem of these tests in general is they are short lasting. They are 5 minutes or maximally 10 minutes long. They depend on locomotor activity. And of course, you always need human interference. You have to handle the mice. You have to put them on a test. They are forced to do something. And all these actions will directly affect the outcome measures. The second thing is that you all know that anxiolytic compounds often exert sedative effects and thereby lower the locomotor activity, which again confounds the interpretation of the results in this test. And this has to be really taken with big care. And overall, we know there is a low replicability of results. And this is what we refer to as idiosyncratic results, where results in one lab cannot be replicated in another lab. Of course, I explicitly say replicate, because with replicate, I mean we want the same outcome direction. We don't have to get the same values, because of course there will be variation. And I will show you that this is not getting better depending on the approaches that you use. Because there's a nice critical review on the open field test, because the open field test is used for 50 years, but there is still no consensus on how to use it. And there are fascinating results on novel approaches, where you see that the open field behavior of animals that explore this deliberately are very different from exploration when you just insert an animal and you test for 5 minutes. This is a nice paper from Hurst and others that show that, of course, just the way how you pick up the animal and put it into an assay will determine the anxiety assessment level. So in this case, of course, people commonly pick up the mice by the tail. There are other methods such as a tunnel. This is a plexiglass tunnel where if the animals are used to move in, they don't have to be otherwise restrained, and you can easily enter them into any test. And we have used this in the past as well. There is the cup, which means you form a cup with both hands and the animals are let's say, habituated to enter this, and then they can be transferred in the test. And as you can see, just lifting the animals by the tail will already reduce the interaction in many lines as compared to the tunnel approach. And of course, the protected stretch attent postures are much increased, telling you that also the procedure, how you get the animals out from the cage, already determine the outcome in your measure. And it gets even worse, because many of these tests are very short-lasting. And here's a nice study from my friend and colleague, Ilan Golani from Israel, who has performed an open field test with 2 different strains. It's the bulb C and wild mice. And what he has done now is, instead of testing for a very short time in a very small arena, he tested for up to 40 hours in a very large arena. And what he could show is that the bulb C mice that in the lab are supposed to be very anxious show a reduced center exploration time in the first half hour of the test, which is already much longer than in many other assays, whereas the time in the open is reduced. And what you can also see is that this is inverted by the with increasing time, in that the wild mice, of course, reduce the center time and stay more at the edge, whereas this flips over from the bulb C to the wild mice. And this is exactly the point, that you can see that depending on the time that you measure, you have completely opposite outcomes. And based on this, the issue is now, what are we measuring, and are the time intervals in which we are measuring correct for the interpretation that we provide, particularly with regard to trading society claims, because this apparently shows that what we are doing is not correct. However, this has not produced any change in the field as opposed to these approaches. I have therefore used a complete different test, which is the light-dark emergence task. And this is a paper from 2006 from a group from Harvard Medical School that nicely report that the way how you start the test will have a huge effect on the variation, and of course also the conclusions that you draw. And I just read this out for you. In our preliminary studies, we found that when started in the light side of the compartment, mice given anxiogenic treatments did not explore rapidly enough to find and enter the dark compartment. Instead, they tended to freeze and remain immobile for a majority of the test session. And therefore, the recommendation is, start out the animals in the dark compartment, contain them for some time, because you can now also habituate them to the environment, so that after the handling they are not too aroused, and then open the door so that they easily get that there is a light coming in, and now you measure the emergence from the dark compartment into the bright compartment. And this reduces the number of false positives, because, as you know, stressed animals with added environmental stress have a cumulative tendency to reduce mobility, and then they will freeze even on the open arm. And this, of course, will lead to misleading results. Another issue is, of course, that most of the tests are done in the circadian phase or in the light phase. So we used and we compared 2 different strains. You see here the C57s and the DBA mice. And I'll come back to this comparison later a couple of times, because we know that the C57s should be the less anxious, and the DBA are reported to be the more anxious lines. And we can nicely confirm this with this assay, in that you see that the latency to enter the bright is almost half of the time of the DBA lines. And the time spent, the total time spent in the bright compartment is double in the C57s than in the DBA. It is not affected by the circadian phase, although there are papers out that show that if you look in more detail, there are differences. And I'll come back to this a little later when I show you our long-term measurements. So this is an important finding, because this tells us, okay, can we compare this with other readouts? Of course, if you now switch over from anxiety, where there is no explicit threat, and we move into the realm of fear, that we can discriminate between innate fear or unconditioned fear, which is fear elicited by, in this case, classically olfactory stimuli. You all know that rodents such as mice have an innate aversion to certain odors. It's the cat odor, it's also the fox feces compound odor. And that means that in the presence of these stimuli, these animals tend to freeze depending on the environment or show avoidance and escape. Of course, it's known that this is a specific receptor. There's a Japanese group that has deleted this receptor, and based on this, you see that there is the avoidance to cats is completely gone, because simply this network does not work, because there is no proper signal detection that would promote the response that is supposed to be mediated by the bed nucleus of the stria terminalis and the extended amygdala. And there are nice papers on this, even very recently. And of course, be aware that, of course, we know that Toxoplasma gondii infections occur in mice. And if you would test these mice that normally have these functional receptors, the innate aversion to the cat odor or cat urine is converted into an attraction. And we know cysts are formed in the amygdala area, but we still don't know how this works. But we know that, of course, also Toxoplasma gondii infection are implicated in human psychiatric disorders. Of course, therefore, it would be wise to look at more naturalistic threats. And there's work from Jean-So Kim at Washington University in Seattle that you should look at that has exploited this. But I now will shift over more to fear conditioning, where I have performed many studies, particularly with fear conditioning, because it's a simple form of associative learning. It's a one-trial experiment that is sufficient to investigate long-term memory. The nice part is that you can, in a single trial, investigate two different forms of memory, context and tone-dependent parts. I think that Jelena probably discussed this to some extent. The nice part about automated systems and fear conditioning systems is that the stimuli and the temporal controls features are completely controlled. And I will show you later, particularly in my second presentation, that the tone can be employed as a very nice conditioned emotional stressor. Of course, the involved structures are extremely well described. We have huge amounts of information about receptors and proteins that are linked in the function. This whole approach is highly conserved across many different species. You can start fear conditioning in fruit flies, but you can bring it up to almost any species, because it's an evolutionary important response. And of course, the idea is that mechanisms of extinction of emotional memories and their disruption are implicated, particularly for post-traumatic stress disorder. And I will come back to some issues where I will show you in a model that I'm convinced that fear conditioning is an excellent model for these basic principles, but I'm not so sure about its translational validity. Of course, the benefit is that if we do this kind of experiment with a single trial, we can interfere in a very time-specific manner. We can interfere before training, after training, before the retention test, after retention test, and do subsequent retention tests. In this case, can be done either with pharmacologic interventions. You know that, of course, we use in the meantime DREAD approaches to target an expression of receptors or designer receptors in specific brain areas, and use then clozapine-related compounds that are specific to be activating these receptors. And in some instances, of course, a lot of optogenetic interference is used by glass fibers in the brain directly, which is also fine because it's excellently working on an exact temporal scale. We know, this is of course a busy slide on the network, that particularly the tone-dependent fear conditioning is a loop from the midbrain to the basal forebrain to the amygdala and back to the output centers, which are in the hypothalamus, for example, for autonomic regulation, in the bed nucleus of stria terminalis, in the nucleus accumbens, and then it moves back to the freezing response in the midbrain. And this loop, of course, for the tone and the shock are separated from the more contextual processing that involves the hippocampus. And therefore, if you use local interventions in the hippocampus. The benefit is that if you selectively interfere locally, you have the benefit that you can use the other form of learning as a control, because this form of learning should have been acquired, whereas you may interfere, improve, or worsen, in general, the hippocampally formed contextual memory. And this is what I show you here. I will show you some data from brain injections where we measured freezing and computerized inactivity as a consequence of dorsal hippocampal muscimol injection. This is a study with a colleague from 2013 where you can see, and this is the reason why I show this to you. Here we have investigated the dose dependency, the site specificity, and also the timing of the drug infection, time infusion before training, to find out how long lasting is this drug. What is the optimal time window to interfere with? And you can see at 4 and 1 hours, we see the most profound impairment, but you see already an effect at 6 hours before, and even 15 minutes before is still effective. Now, the dilemma of pharmacological injection is that we don't know exactly where the drugs are really acting, because we are providing them in the dorsal hippocampus. The red dots indicate you where we have injected, but we actually don't know whether they are specifically active only in this injection area. And this is a particular dilemma in highly lipophilic compounds. So in this case, what we have used is now a fluorescently tagged compound, which is muscimol poly-P, to see its distribution profile after injection and to follow its distribution thereafter. So what we found is there is leakage based on the cannula to some extent, but maybe because when we pull out and terminate the animals and pull out the cannulas, there is some leakage. But we can see that the distribution is roughly contained to the dorsal hippocampus, and the highest level of binding is in the areas where we know that GABA receptors are expressed in the hippocampus. And we know that 24 hours later, this fluorescence is gone. And based on this, now we know exactly that this depends on the molecular size, the lipophilicity, and its in vivo half-life of the compound. And you have to be aware that although we nicely did this for muscimol, it is not done for many other studies. And I want to take studies for granted that say drug injection in the intracerebroventricular area, which would be roughly here and here, would mediate hippocampal effects. I have published this year a study with the neuropeptide galanine, where it's clear that the ICV versus the dorsal hippocampal injection has completely different effects on the behavioral consequences. So in the beginning, I told you we have used studies with fear conditioning, and I will show you old data from 1999, where we could nicely show that despite the fact that the DBA mice are very anxious, as I showed you before, in fear learning, they are very poor. And it's the C57 substrains that are really doing well. Yeah? And therefore, in many studies, the DBA mice have been stamped as being hippocampally impaired. And I will show you later that this is not the case, that there's something else going on. So an alternative paradigm that I have used, particularly when I was in Sweden, is the passive avoidance paradigm. The passive avoidance paradigm is nothing but a modified dark light box test where you place the animals in the bright compartment, and they have the urge, particularly if the light intensity is very bright, to move over into the dark compartment. You can confine them in the dark compartment, and then you can expose them to a shock. And then after defined time interval, again, you can test, are you deliberately entering the dark compartment? And if the animals have formed long-term memory, they should inhibit to move over. And that's the reason why it's called passive avoidance or inhibitory avoidance. The good part here is that in fear conditioning, the animals freeze, but they don't know actually what the threat is. Here they know that the threat is the door. And I will show you later that I have used other approaches where exactly this turns out to be the most important aspect, that the avoidance is directed to entering this compartment through the door. So we measure step-through latency. And I'll give you some ideas here. Of course, the nice data on 5-HT1A receptor agonist shows that if you use low doses of this agonist, you can facilitate learning. And with you use higher dosages of this compound, which is supposed to activate postsynaptic 5-HT1A receptors, you will result in a massive impairment of learning and memory. And therefore, this assay has a better chance, particularly if you titrate out, and I will show you this later, the shock intensity, to use it in both directions of improvement, but also impairment. The same applies here for a 5-HT1A receptor antagonist. If you use it, you produce dose-dependently and facilitation, and we know now that this is partially mediated by the fact that the overspill of the endogenous serotonin is binding to the 5-HT7 receptor. So this is very interesting, and the nice part is that this is partly work done by my colleague in Sweden, and I have always replicated his findings in my own lab, whether we used mice or rats, different assays, different housing conditions, we can replicate the findings, which is very important. The last thing is, of course, you can use also a muscarinic acetylcholine receptor antagonist like scopolamine, which of course are used for models of cognitive impairment. And you can nicely find as well that here you see a massive impairment. So I have transferred this again to DBA and C57 comparison, and while the training latencies are similar, there is a little difference, but these animals have been habituated to this. You can see that in the retention latency, the DBAs are always performing on a lower level, as compared to the C57s. So we can confirm in the classical test that the DBAs are inferior in learning, despite the fact that they are having a stronger anxiety response. We also are able to look at the repeated level of freezing after re-exposure to the conditioning environment based on contextual cues. And you see the C57s stay elevated for longer, and the C57 BL6Js extinguish well. And you can see here that in the tone-dependent freezing, it has a similar function. Actually, if this would hold, we should see, and this is confirmed here, that since we know contextual dependent freezing is similar to avoidance, because it's also a contextual part, we see that with increasing shock intensity in the subsequent retention tests, and this is the C57-6J and the 6Ns, we see a subpopulation that takes longer to move over and does not extinguish as fast as the C57's BL6Js. And I will show you data where, surprisingly enough, despite the fact that we have the conclusion that the 6N mice show a PTSD-like phenotype, in other classical tests that never emerged. Now back to the comparison between DBA and C57. Many people use the so-called water maze test in order to look at spatial orientation. We have developed a new assay, or an assay in almost 10 years ago, which is the modified Barnes maze. So we have a lot of holes, and we always select the target hole in the intermediate ring of the holes. If we use negative reinforcement, meaning wind and light only, you can see that the DBAs are much slower in finding the escape hole. The distance moved is increased, the velocity is the same. If you do a probe trial and you remove the target hole or escape hole, you see that the C57s explore the target area with higher preference than the DBA. And that confirms what we have all written in the literature. However, we used now an approach where we just added one positive reinforcer, which is an element piece. So we provided all holes with element pieces that were not accessible, except for the escape hole where an element piece was accessible. And under these conditions, there is no difference between these animals. On the contrary, you see that the DBAs are motivated by the element piece as a positive food reward and increase their velocity. And in the learning trials, there is no difference. So the conclusion that we had at that time is, since we know that the DBAs are more anxious, has it to do with cognition or has it to do with the emotion impacting on cognition? And this brings us to a very important principle, which is the stress impact or the arousal impact on performance. And this is referring to the Yerkes-Dodson law, which originates in 1908 and is well known in psychology. If we do simple tasks and there is a low level of arousal or stress or motivation, there is a low performance and you reach a saturation. If you have a difficult task, you reach a saturation, and then if the stress becomes too much, the performance goes down. This is a paper from Diamond. And of course, at that time, you can nicely see fear conditioning is listed here as a simple task. Whereas, of course, we know that fear conditioning, particularly the contextual part, should be classified as a more difficult task because we know it's a hippocampal dependent task. And of course, this is the issue why, of course, we cannot differentiate now. Is this effect, or is the performance now affected by the anxiety state of the line by itself? And of course, can effective or unspecific stressors impact on the cognitive tests? And I will show you in subsequent tests where I come back to this, that this is the case. So what we in the meantime have developed, and these are examples of recent approaches, is we have started to develop home cage-based phenotyping. That means we get away from handling the animals. And the classical example is one of the first papers that I've published on this, is that we have a home cage connected to the test compartment. It looks like an automated passive avoidance task, except that the animal is now living in this compartment. And we are using different outcome measures, and we are looking at novelty in the beginning. We let the animal habituate to this environment, and then we do training, and then we do a retention test, which lasts for, in this case, 2 days. The good thing is that, of course, the experiment comes to the animal. The animals decide when and if to participate in the test. And the timing of the tests is exactly coupled with the peaks of activity. So thereby we avoid any unspecific stressors. Yeah? And this is the paper that we published on this, and there's more to come. So let me show you some data, and watch out, there are very different aspects on the time scale. First of all, if these are the transfer latencies we see between PLEC 6J and 6N mice, there is no difference in the training phase. This is an inverted Kaplan-Meier, where you see this is the time where 100% of the animals have transferred into the test compartment, and this is the video where you can see the same for the others. There was one animal sound asleep and did not notice that even the door was opened, and the half time is the same. Now, if we do the retention test, and we use animals that did not receive a shock upon exploration of this test compartment, you see the half time to move over is reduced. They are over in half a minute. The only motivation to move over is novelty seeking. If you use now animals that have, and retest animals have been exposed to the shock in the training session, you see there are substantial differences. Now, first of all, you see a proportion of animals moves over relatively quickly, but quickly is now relative because this is a time scale of hours. So the first animal moving over will be beyond any time in any of the passive avoidance tests being used under the same conditions. So this tells us that, of course, the behavioral responses in many passive avoidance tests are hampered by many unspecific stressors. The second thing is you notice that the animals are only, let's say, making decisions in the dark phase and not in the light phase. And you see that some animals are not moving over at all, and this is shown by these arrows. However, you see a massive difference in that the B6J mice that are tested are much faster to move over than the N mice. Yeah? And this is, of course, confirming some differences that we saw. Now let me show you a movie where I show you in a time lapse the activity of a mouse in the home cage 5 minutes before the door opens, sped up 10 times so that you can see what happens when the door opens for the first time. So you can nicely see the animal is exploring the whole cage. It is grooming. There is food on the bottom. There is a nest. And the animals do not show any sign of hectical moves, risk assessment whatsoever. And the animal does not mind to be close to the door. Now the door opens and you see the animal moves far away. It approaches the door and it peeks, and it does that in a stretch a tent posture-like manner. And this is the classical part where we now use computers to completely quantify this 24/7 with even body length measurements. And this tells us that, of course, if you just look at this, you realize the open door is now a threat. The fear is proportional to the magnitude, or the distance to the threat is proportional to the magnitude of the threat. So we have a problem. We have too small cages. And I always say, probably you are testing Ferraris in the garage. We should have a larger test, because this is known in ecology that this is a very important readout. So now let's take a look at the exploration. And this is a graph where you see the non-shocked animals in the home cage move back and forth. This is their position relative to the door or the test compartment. And once the door opens, they move back and forth immediately. And if you look at the body length, which is the unfiltered signal, there is an intermediate body length all the time. In the shocked animals, you can see this is very similar in the beginning, but when the door opens, the animals move back. And then they have these spikes downwards to the door, where you see after 10 minutes, this is the first time that the nose peeked into the test compartment. At the same time, the body length becomes spiky in that whenever the animal approaches the door, the body length increases, which is a stretch attent posture, which is nothing new. It's described since 1963, but we quantified it, and we can now nicely see that the steepness of the slope of the body length versus the distance to the door is a nice indication of the fear. And if we would continue this in an animal that would extinguish, this slope would become 0 as soon as the animal extinguished this fear. And we've measured the slope fit during the training versus the testing, and in the training it is not there because the animals are not afraid yet to enter. Of course, there is novelty seeking, but there is no difference. Now comes the second level on this. We tested now what is happening if we give different reinforcement strength, different shocks. And we also included, that was initially a software mistake, a late shock group. The late shock group is a group that was enclosed here and shocked in this compartment 1 hour after it entered. So this is a latent inhibition group. This group shows a significant increase in the latency to re-enter, but in the time scale of minutes, whereas, as you can see now, animals exposed to 3 shocks compared to 1 shock perform similarly, but animals exposed to 5 shocks move over relatively rapidly, and there is no persistent avoider anymore, telling us that the reinforcement strength of 5 shocks impairs memory. Yeah? And we have a ranking on this, and we can see the same thing in C57BL6N. Remember, they were stronger to begin with. With 3 shocks, they are already weaker in their response. And with 5 shocks, we see animals that move over relatively fast, and the other portion never goes back. So these are the persistent inhibitors. So now this makes us believe that since this is a time scale of 2 days, and just an indication, here is a single response where one of the few occurrences is that an animal made a choice in the light phase. This tells us that we have to look even at subpopulations of individuals in genetically identical animals that show a PTSD-like phenotype, telling us that this is epigenetically determined. And this is something that we have to be aware, and therefore I have done a review. There is no fear conditioning study that shows any form of impairment, because the higher the shock level, the more the unspecific freezing, and therefore this is not related to the fear association, but to a general increased anxiety of the animals based on strong punishment. It's like behavioral suppression. Yeah, and this is of course an issue. Now what is interesting is that in the classical tests we showed that there is an extinction, and we know that the animals that are moving back explore the test compartment in one go. And we found out that we had a correlation that the later the 6J enter this compartment, the slower they progress. There is one here really flat down in order to explore this compartment. Now what we know is, of course, since this is a cumulative plot, if there's a flat line, there is no progression in the light phase, obviously. However, if you check the 6 ends, you see there is a delay in entering, but the progression to re-explore and revisit is unchanged. So I start to doubt what we are actually measuring when we are measuring extinction in the classical fear conditioning approaches, where we use a lot of unspecific stressors. Yeah? And this is very different from the classical tests, and I have confirmed this in other studies as well. So now let me go to the last part. Now we checked again the DBAs versus the 6Js in this assay. And it turns out that in the training, yes, we see a difference. The DBA are more anxious, which classifies as neophobia. However, this is already gone in the non-shocked animals when they re-enter the second time. There is no significant difference anymore. And if we do the retention test under these conditions, the avoidance and everything is completely preserved, and telling us that the interpretation that we have, that they are poor learners on aversive reinforcement levels, does not hold. Yeah? We see the same large inter-individual variation, and based on this, we think we have to discriminate, and we should beware if we use one, let's say, reinforcer, we may not be able to compare different lines, because we have to test them with different reinforcers to find out how their stress susceptibility will impact on the cognitive function. And we have to correct this whole approach, and that's what we are currently preparing in our studies. And therefore, if we continue in the classical test range, I'm afraid that we will draw a lot of incorrect conclusions and about what is the real performance and what is the contribution on emotion, on cognition, and on emotional expression itself. And actually, I have confirmed this lately with another animal model. So we used an Alzheimer model, the APPS1 mouse line, which has been reported to be learning impaired in the Morris water maze test at the age of 4 months. We used an, let's say, figure 8 maze, completely automated, connected to the home cage, in order to find out whether there is a symptom progression telling us something about cognitive decline. And we could not find it, because lo and behold, the C50, the APPS1 mice are also known to be very anxious. And the water maze is the least suited test if you have a high anxiety like phenotype to test mice. And I think in general, it should be banned as a spatial learning test for mice anyway, because of its negative impact on many functions. So my conclusion is now that we have home cage-based approaches that are substantial refinement of behavioral phenotyping. We can use unambiguous behavioral measures, particular avoidance is very useful because that is part of the DSM-4 criteria, and freezing is not really useful in this case. We can show the impact of negative stressors on emotional responses because we have contradictory results. There's a nice paper from David Wolfer and Hans-Peter Lipp from 1997 showing this already, but drawing the wrong conclusion. And of course, it's important because we want to replicate and we want to translate research which is not specific to the lab, so that the credibility crisis that we currently have can be overcome. And we are really intrigued that we are moving in this direction, and this is a cost or EU proposal where a lot of groups in Europe are working together because we are thinking about how can we make this possible in the future, what are the standards, and how can we develop this further on, because we think this is really important. And we have to stop behavior tests that have no added scientific value and only unnecessarily compromise animal welfare. And this is the topic that is still running till the end of the year. So we have already 18 articles, and it has drawn a lot of attention. And I have to thank a lot of people that helped me in all of this, because we had the benefit of an excellent fine mechanics and electronics department. We had a lot of partners. And of course, there were a lot of contributors. And I thank you for your attention and look forward to your questions. So let me stop sharing. Oh.
Thank you very, very much for a very interesting talk.
Rajneesh, it's first part of Sir lecture. Sir has got one more part.
Okay. So maybe we will we'll open the discussion after the second part, right?
I would prefer if we maybe discuss a few issues now, if you don't mind, because the behavior part, of course, and the second part is a very different one. I will give you another twist to the story.
Okay. So, as per Professor Oliver's suggestion, we can have a small 5-minute discussion session about any queries, questions, or concerns. The floor is the session is open for discussion.
Sir, I have got one question. For any stereotaxic dependent studies, brain atlas is a prerequisite to know the coordinates. Can you please comment what are the mammalian animals for which this brain atlas are available, and is it age specific or it's a universal one?
Actually, to be honest, I have used a mouse brain atlas for stereotactic surgeries, but I have to warn you, because most of the tissue presented in the atlases is shrunk tissue. So if you want to really do local injections in vivo, you should try out first with dyes where you exactly inject. And then you have to be aware that there are differences also in the thickness of the skull of some mouse lines. And I can tell you, you should contact me. I have a list where I would give you coordinates, and we use for example, for pharmacological injections, we use delicate spacers to titrate the exact depth, for example, to avoid that we damage the dentate gyrus in the hippocampus and so forth. So this is very delicate, and I urge you, if you want more details, send me an email and I can give you all the details that I have, because it's a delicate one. I have probably done, I would guess, 3,000 surgeries to very delicate brain areas, and you have to have a good stereotaxic system. There are fantastic holders, but you have to watch out because the age, the skull thickness are all determinants. And of course, be aware, if you use very old animals, aged animals, you will have an excessive loss, despite the fact you do the same procedures as you do, for example, in 10-week-old animals, there is no loss. So there are a lot of tricks and issues and details that I could answer. I hope this answers your question, because I could not understand everything. Hopefully that was correct.
Yeah, that is fine. That means the standard brain atlas that is available in the market, that is not age dependent. It's a universal one.
There is a universal one that is no age dependent. Also, the one that I've used from Watson and Paxinos is not age dependent. Yeah, but as I told you, we have developed our own stereotaxic systems where you put a drill on the brachma, and then you have the option to use move in the dorsolateral, or let's say in the lateral direction and the frontal caudal direction with micrometer screws, and the drill is fixed. The drill is fixed on a holder so that you can really drill perfectly. And we use it now for viral injections, for optogenetics, everything, and it works really well.
So you perform all these experiments with respect to brachma and all?
Yes, we do it. And of course, you have the option to also tilt the skull so that when of course you sometimes have a skull that is not really nicely leveled, you can tilt the skull so that it is on a proper plane, because that's important.
Okay, sir. Okay, thank you, sir.
Yeah. Actually, in the past, I did courses where I invited PhD students to learn about this, but since I have so many now administrative tasks as well, I have stopped this. So if you want details, let me know and I can send it to you.
Thank you, sir.
Yeah, you're welcome. More questions on the home cage-based phenotyping? If not?
No, I think it's a new next talk.
Yeah. Okay, then I will move over to my next talk.
Yes.
So let me again entire screen.
Sir, before you proceed to the next talk, I would just ask a general question.
Yeah.
As you were talking about fear conditioning,
Yeah.
so it's our general observation whenever we move in dark, and generally when we are alone, when we are in a group, we generally don't develop fear. But when we are alone, we do develop fear. So what is that something that goes inside the brain?
Of course, be aware that the fear, probably, I don't know, I guess Jelena Radulovic talked about this. If you have group housed animals, it's very obvious that your fear response will be attenuated based on, let's say, the interaction with conspecifics. Yeah. So, and also, if you have a non-conditioned animal, this animal may have shown increased anxiety-like behavior because there is emotional contagion. The next thing is you have to be aware, we do often studies where we do fear conditioning as a test at the end of many other tests. And then you will be amazed that many mice do not acquire fear conditioning anymore, because the foot shock is for them not something to be concerned about, because they've made a lot of experiences. And I think what I have published in the past is that I said that many mice that are naively brought into fear conditioning over-respond with their fear because they lack essentially experience. So if you would use experienced mice, the fear would be much lower. And this is exactly what I showed you in the latent inhibition part. The animals have been exposed to a shock. They have a very strong response to the shock, but they do not associate, and therefore there is no avoidance response to the conditioning chamber. And that means, of course, particularly when it comes to animal welfare, a lot of people have said in the past that we have to watch out because we are using a traumatic stimulus. This is complete nonsense. There is no traumatic stimulus. It depends on the experience of the animals, and most of the animals that we are using are relatively unexperienced in an impoverished environment, and that does not meet the criteria of what we should be dealing with.
So does that mean that as we age or we become mature, we become more fearful as compared to the newborns?
No. As compared to unexperienced animals, we would become less fearful, but we are better in discrimination what is a real threat from what is not a threat, because the mice in the beginning are so unexperienced that they will consider everything a stressor. And I've published
Why I'm asking this question is when my daughter was small, and
Yeah.
she doesn't have this knowledge of all these things. So when I used to take her near the balcony, I stay on fifth floor. So she never used to react. Now as she is growing, now whenever I take her near the balcony wall, she starts having fear. So is it that with age we develop the feeling of this fear and all? And when children are very small, maybe 6 months or 1 year, they don't understand all these things and they don't have the fear.
That is of course correct. Look at if you look at which animals fall victim to predators, it's the young one, unexperienced, because they don't show fear, and it's the older animals because they are not physically fit and cannot compete again. So what that means is that, of course, also, especially humans, show a different, let's say, way of responding to the environment. I can give you exactly the same story. My daughter had a mouse. It had cancer. I asked her, if you will assist me, we'll do the surgery on the mouse, and then it will recover. And she did. I guess nowadays she would not do it again, because if she would see all the blood and stuff like that, she would not like it. Yeah, it's the same issue. There are preferences, developments, which of course change how we see certain things, and of course a lot of people move away from doing certain things because they now understand all its implications.
Okay, thank you, sir.
Yeah.
Sir, may I ask you one question? This is Arunima. Sir.
Yeah. Hello?
Hello, those individuals who are into adventure sports, do they experience the same thing every time? Do you have any evidences that whenever somebody is doing a bungee jump or paragliding or parasailing, the first time, whenever we are exposed to certain conditions, we get fearful? Is it a conditioning that later on it becomes a habit and we don't, we are not frightened anymore? Is it like that?
There is a contributing factor, but this is again individually different. So I always talk to the students and say, what is the most ethically approved stress test in humans? It's public speaking. And as you know, of course, it's not about public speaking. It's public speaking about a topic where you know that you don't know, to people that are supposedly experts. And this is the most challenging ethical test that we are allowed to do. And of course, you can imagine that the people are really having a high stress level. Their anticipatory response is already pretty bad, and it's a severe stressor. So what do you learn? You learn to deal with situations. I'm still aroused when I start to give a lecture, but I'm calming down very fast because I have a lot of experience. I teach a lot. I teach sometimes in 3 languages. So this is not an issue. But for a newcomer, this is a difference. And of course, there are people, I'll give you an example. There are scientists that have been really fantastic in their scientific career, but these days they would hardly survive because they have been unable to give a lecture, because they are not able to speak and, let's say, promote information properly. And there are differences. And I don't know how this can be changed, because I think, and I've started this, we should also see our mice, and if we enrich them, you will see that they are tolerating things much more easily. And that's the reason why I come back to what I mentioned before. There are individual differences where some people and some mice get sensitized, they become more afraid, and some become less afraid. We don't know how it works. So I have been discussing with colleagues, what we need is, I always make a joke about it, but it's like in Men in Black, you know the movie?
Yeah.
Where you need the flash to reset the memory of the same animal, test it over again, and see whether the same outcome holds true. We don't know. But we know that if we have a very careful animal, and we'll test it in several different tests, and this has been also reported for the cardiovascular system, this show the same direction of, if you have a cohort of 10 animals, the animals that is the strongest responder in test 1 is very likely also the responder in all the other tests that are being conducted. Yeah. So there is a bias.
Yes, sir. Please continue.
Yeah, there is a bias where there seems to be epigenetic trait. And in humans, the classical situation is the first child is always the careful, the second is less careful, and the third child is the roughest. It has to some degree also to do with the maternal paternal care, because our first child is very valuable to us, so we are very careful and caretaking. Also, we are less experienced how to deal with our children, and that is reflected in many human studies as well, and I tell you it applies as well to mice and their offspring. Okay. Did I answer this? Is this working?
Sir, it's fine.
Or am I out? Okay. Then let me, okay, now I will, so now I will move to the second part. I hope you can understand me. And now I will give you some ideas about behavioral and pharmacological interventions on heart rate dynamics, from physiology to pathology. And in the second and last part, I'll give you 2 examples of EEG measurements in freely moving mice. And to speed up a little bit, I have a great experience in so-called radio telemetry. The disadvantage is it is an invasive procedure where we implant animals with a micro device to measure, as you see here on the top left corner, an implant with a battery, electronics, and the subcutaneous electrodes, which are in the end, let's say, de-isolated at the red spots so that we can measure the heart rate in the mice in the home cage. And I will show you that all the studies that I have performed are done under home cage conditions. And we also have optimized pharmacological intervention studies that are done under brief anesthesia under very refined conditions to make sure that our injections do not interfere with the consequences. The good part is we have done many of the studies, particularly in the light phase, because then we know physical activity does not contribute to the heart rate changes that we are measuring. Yeah? And this is something that I will give you some idea about. So remember the fear conditioning I showed you before. This is also a very old study. That's actually how I got started before I moved from insects to mammals. Here we have fear conditioning where a tone was presented together with a shock as in paired. It was separated by 30 seconds, or it was separated by 60 seconds, or we had only a shock or only a tone. And we did this in the fear conditioning box, and then we measured in the home cage what is happening in these mice when they're hearing the tone. And what you can nicely see is the baseline heart rate of the mice is in the range of 550 to 600. And once the tone gets on, the animals that have been exposed to the paired tone and shock, or the 30-second unpaired, which is trace conditioning shock, show a massive acceleration close to 750 on average. And then slowly during the tone presentation, it falls off again. Here is a classic example of an individual. The tone gets on and you have this massive increase. If you measure the blood pressure, it comes with a delay. If you would measure a paired training and you give only 10-second shock, the heart rate will still accelerate after the tone is turned off, and it will take time to recover. So if I see fear conditioning studies where they measure freezing to a 30-second tone and then freezing to without tone and then again with a tone, this is nonsense, because the autonomic system is so primed that it is already up and accelerated, and then it will move back and forth. Look how long it takes till in the paired mode, even after the offset, the animal is back to baseline. So this tells you, and you know this, if you're under severe stress and if you are, let's say, in a dangerous situation, your heart is pounding. Yeah? So now we can do curve fitting and see that the half time to reach the maximum is about 3 seconds in heart rate, and in the blood pressure, it's more than 50 seconds. So this is a very fast autonomic response. And we also, I forgot, we also know that if I would now replay the heart rate response day by day by day by day, it will fall back to this level as in no shocked animals. But however, this is still a significant increase. But it tells me, and I can tell you it almost is a joke, but it should not be surprising. I have tested mice that show no response to the tone whatsoever, simply because they have been deaf. Or in many studies, the wrong tone has been used. The tones that we use are 10 kilohertz, because that is what the animals are up for. And what we used extinction of heart rate responses in the home cage, what happens is that even the black 6Ns that show a delayed fear response in the classical tests show the same fast extinction as we have seen before in the home cage situation. So extinction is a very nice autonomic measure as well of the fear expression of these animals. We have tested some mice lately, and this is published in Molecular Psychiatry, where in combination with a colleague, we measured the P11 mice. The P11 is a protein that is highly expressed in the brain areas linked to depression. And what we realized is, I would not call it depression at this moment, I would call it that whenever we put the animals in a novel situation, they have a higher heart rate despite there is no weight differences than the wild type controls. And as you can see, they take much longer to recover. This is the heart rate drop off. So look at this. If you put an animal in a new situation, it will take half an hour to an hour before heart rate even reaches baseline. In the home cage, we have measured up to 2 hours. Here you see the fall off of the heart rate variability. The heart rate variability in the wild type is higher, but it falls off stronger because it increases higher. So we tested the animals in fear conditioning, and oddly enough, this is the one and only strain where we already saw what we do is we place the cage under a loudspeaker. Just the transfer arouses the animals so much that their baseline is elevated to begin with, which is of course not nice because we would like to start out at similar conditions. But there is no way, it takes them many days before this is almost gone. So day one, you see, this is the tone response. The wild types are accelerating. These are even higher, but they recover much slower. Look, this is the fall off, as you can see, extinction in the wild type controls after this amount. Here, look at the range. This is now a very small bump. This is only roughly 100. And the maximum is in the range of 550. Whereas the others are still higher and it takes them longer to recover. So I'm not sure, for me, this is more like a stress response hyper-responsiveness and the delayed recovery to baseline. Now we were intrigued to find out what are heart rate values, what can we measure, what can we compare. And we have tested a lot of pharmacological interventions. I will not go in details, I will highlight some of them. And we recorded for 18 minutes, which yields roughly 10,000 beats. And we used this in the end as a side project because we found out that, of course, many of the drugs that would be argued to interfere only with cognition, of course, also interfere with the autonomic system. And we cannot separate. I'll give you some examples on this. Now, if this is the heart rate and we rank this according to maximum from high to low, we see novelty is higher than restraint. Take a look at this. 800 is roughly the maximum physiological limit. This is the control value indicated here in gray. And then this is sleep. These are different pharmacological interventions. I will show you in a minute more details. And this is anesthesia with ketamine Rompun. Not surprisingly, the standard deviation of the heart rate or its variation is invertedly related to this. If the heart rate is high, the heart rate variability is low and vice versa. Of course, there are some outliers here. And you see that, of course, certain compounds have different effects. And now we can rank them and we can compare what this how they are related to each other based on those two parameters with similar rating. Now, there is one problem that I would like to expose you to, which is very important to note. So if we look at heart rate as a function of time, and we would cumulatively measure linear measures such as the mean heart rate or the standard deviation or the root mean square subsequent difference, we see drift-like behavior. This is the heart rate in its temporal sequence. And we see drift-like behavior except for the root mean square of subsequent differences. So this is what we call to non-stationarity. It's a physiological measure. So if we would now sequence randomize this, meaning we would reshuffle the sequence, we still would measure the same mean. Now these individual information follow exactly the rules that we need to apply linear statistics. There is in the beginning a little drift of the data, and then they merge at stable values. So that means here under these conditions, it's only the RMSST that provides for stable values. The others are continuing to drift whatever we add. The longer the time interval, the drift never stops. So the question now is, should we then use this? Because if this is the case, what is the basis for this? Now the basis for this is if you compare this pattern with itself and you perform an autocorrelation, it will be identical to itself. And if you would compare it with a shifted pattern shifted by 1, you will see that there is a high degree of correlation which only fades off into a range of not correlated if there is a shift of more than 600 beats. And that means there is internal correlation that will provide information on the status of the system. Not surprisingly, if you look at sequence randomized data, the autocorrelation is gone with a shift of 1 beat. This prompted me, together with my colleague Michael Meyer, to publish in 2003 a nice article when we said, when the mean is meaningless, because now if you look at these data, they don't tell you anything because the means and the standard deviations are the same, but the physiological characteristics are completely different, as you can see. And this tells me, oh, there is something wrong. What can we exploit now? So what we now think of is conceptually, if we have heart rate fluctuation as a function of time, this is the RR interval in milliseconds as a function of time. The question is if we can extract information where we can predict where the next beat will come. And this is like forecast. And if this holds, can we measure whether we can predict a pathological state where this is not possible anymore? This is like weather forecast. As you know, under certain conditions, you can make a nice prediction. Under certain conditions, you cannot do a nice prediction. Now, there is an analysis form called detrended fluctuation analysis, where I don't want to bug you with all the mathematical details, but what you can measure is a so-called detrending analysis, where you look at the fluctuations driven by stimuli. And you can detrend this. This is the formula. This is published in Chaos in 1995. It's a unifractal nonlinear approach. And if you do this, what you find out is if you look at the logarithmic, the decadic logarithmic of the fluctuation of the beats versus the number of the beats, you come to a points that can be fitted into a line, and this line in a double log block, this linear line, only in a double log block, will tell you something about the internal correlation of beats. And these are the extremes that we can find. We can find an alpha of 1.5 Brownian noise. We can find persistent long-range correlation, which would be pink noise, which would be the red line. We can find an alpha of 0.5, which is white noise. And now the question is, what do we measure? And we measure in normal animals, and we find that during sleep, in a wake state and aroused state, we measure 1. Persistent long-range correlation of the heartbeat interval fluctuation, whereas during the fear state, there is a removal. It drops down to 0.5. What we also see is that in adults, in humans, we have 1, and I'll show you more data in a minute. We know in pigs it's 1. We know in dormouse awake it's 1. We have tested Siberian hamsters, it's 1. And that means, oh, this could be a very useful measure to identify the normal state of dynamics unrelated to the absolute heart rate. And so we use these kind of approaches to look again at the pharmacological effects. And what we found out is, if we use at the alpha fast, that means the short-term correlation in a data stream, we find out, oh, there are compounds, and this is our vagal inhibitors, where you see the short-range correlation is moving into more Brownian noise, 1.5. If you have high vagal hyperfunction, and these are partly anesthetica and other compounds, you have a vagal hyperfunction, it becomes chaotic, random. And this is, in the end, overlapping with the alpha slow value, telling us that we are able to identify pathological states of dynamics. And if you do the clustering, it looks very different from what we had seen before. And I will show you some data on 8-hydroxy-DPAT in a detail later. So if you compare the two approaches, the linear measures, where we have on one edge sympathetic system overactivation and parasympathetic system hypoactivation versus exactly the reversal, and we compare it with the nonlinear measures, we can argue the extremes are pathology and the center is physiology. And if we would look at the ranking, oh, the ranking looks very different. I'm now getting worried, what the hell are we doing with the linear measures? Because they are not very informative. And this is an issue that, of course, we have to be aware of. So let me take an example. This is 5-HT1A receptor activation that results in pathological heart rate dynamics. So we use 8-hydroxy-DPAT, and what we normally see is, remember, I showed you this in the beginning, this is your baseline heart rate. And now I have an extremely extended range, and this is the tone-induced tachycardia. Our 8-hydroxy-DPAT already changes profoundly the baseline. It's moved downwards, and it's very spiky. So a lot of gaps in between, and then accelerations again. And then we have a very brief acceleration. The fluctuation is extremely high. And many of these 5-HT1A receptor agonists have been described to be anxiolytic. This is not anxiolytic. We know now that the baseline effects, the blunting of this, let's say, reduces the responsiveness of the system. This system is clipped and clamped at 600 beats per minute, roughly. Now the dilemma is, many people say, or the scientific community, it clearly expresses decreased heart rate variability is bad and increased heart rate variability is good. And what I can clearly say, that what we measure here with all its symptoms is mimicking the side effects of selective serotonin reuptake inhibitors like citalopram, where a warning came out that this can result in cardiovascular side effects. I published this paper and I told, and I clearly expressed pathological heart rate dynamics. It took me 3 revisions and 150 queries before I had to ask whether someone of the referees was from the pharmaceutical industry, because they didn't want to hear that there is a negative effect of SSRIs on the cardiovascular system. And there's a big debate about this, because it turns out that we have ourselves found out that the so-called silent SSRIs are not silent at all, because they blunt often responsiveness to external stressors. So the question is that now beneficial or is that now detrimental to the system? And I think that if you have a compromised autonomic function, it could be associated, because there is an association on one hand between neuropsychiatric disease and increased cardiovascular problems, but there's also an association with the treatment. So what are we talking about? It's the chicken and the egg problem. So now to move on, we have tested adults under different conditions because we wanted to know more details, and we can say that this is clear that adults in different conditions have roughly one. It's only at nighttime when there is a vagal overactivation that this sinks down to some extent. We know also now that people with congestive heart failure have a shift in the direction of more Brownian-like noise. And if you completely have a heart transplantation, meaning the neuroautonomic control is gone, you have Brownian-like noise. And we know that there is recovery longer after transplantation, where there seems a return of neuroautonomic control occurring. Actually, we have also data from another clinic where this does not occur, despite the fact that theoretically the surgery is done the same way. So now we think, that tells us that our scaling values can be used and can also be applied to humans. So we applied this, and this was mainly done by Michael Meyer, to a study of humans where these are the so-called tachograms. This is the heart rate interval fluctuation as a function of time, with the RR in intervals in milliseconds, where this would be a relatively tachycardic patient, and you see the patterns look very different. Based on this, and what we should add on is there was no effect visible in the ECG signal itself. It was only that the tachogram looked different. Based on the scaling, and that was blinded, we could find out and determine that in the patients where we now would argue the scaling coefficients are completely deviating, and we have in the meantime split this up into fast and slow, that where the scaling coefficient is deviating, that here we see an autonomic problem. And of course, what happened is we have an excellent diagnostic, but we don't know what to do, because the point is it comes from the central autonomic system. We, in the meantime, have used these approaches for the first time in this paper, and thereafter again, where we could nicely show that PTSD patients seem to have a diminished tonic parasympathetic activity during the sleep phase, which distinguishes these from the normal patients only in the night phase. Yeah? And this is a linkage where in the past people have thought, okay, affective disorders are associated with an increased sympathetic drive, and we now think it's more a decrease in the parasympathetic tone, because you all know that heart rate normally is under constant tonic parasympathetic inhibition as the fastest component, and particularly during sleep, this is important for recovery. And therefore, what I want to point out to you is that, of course, that we are now using particularly these irregular heart rate interval fluctuation, or the dynamics or the noise, as a nice index of the proper brain-heart interaction. Actually, I think that this would be the future to go also for safety pharmacology of new drugs. We think that the nonlinear methods are superior to conventional linear methods to assess qualitative differences and to identify pathological states and distinguish them from physiological states. And we think that this now allows us to look at central autonomic dysregulation under pathological conditions such as epilepsy, with a true translation between species, including humans. And with this, I think, of course, I showed you that, of course, we now talk about mainly attenuated parasympathetic function in PTSD. And of course, you all know that, of course, relaxation aspects and breathing in and out can stimulate and recover some of these functions, because in particular the breathing cycle is heavily coupled to the autonomic output. If you breathe in, in a relaxed state, your heart rate will accelerate, and if you breathe out, it will decelerate. And this respiratory sinus arrhythmia, which of course has nothing to do with any pathological state, is apparently blunted in people with depression and other affective disorders. Yeah? And now, based on the epilepsy, I will now give you some ideas on this aspect, because the telemetry systems can nicely be used for this. And I will give you an example of two models that we have used based on scientists that have discovered the genes or have worked with the genes that have been implicated in this disorder. The first one is the so-called Ohtahara syndrome, which is an early infantile epileptic encephalopathy. The point is that children that have a mutation in this gene, which makes at least one of the alleles dysfunctional, have this. And of course, this is related to, in the beginning, delayed development and epileptic symptoms, and of course, profound motor and cognitive defects and very poor short lifespan. It's a really terrible situation. And the second one is megaencephalic leukoencephalopathy, which is MLC. There is a group in our university working with these patients, which is a form of vanishing white matter disorder. The vanishing white matter disorder is a disorder where patients have a decline. It's often picked up by imaging, decline in the white matter, which is a demyelation. Of course, you can easily imagine that there's an increase in excitation because the neurons are not isolated from each other. It also is associated with seizures. It's associated with a decline in motor control. And often it is not discovered before a trauma occurs where the disease seems to be starting. And I'll show you some examples where we have started to work with these models to find out what can we get in these models, what do we see, particularly with regard to the EEG recordings. And in the MUNC18 or the syntaxin binding protein 1, which is crucial for presynaptic neurotransmitter release, the issue is that we saw spontaneous epileptic seizures, stroke tail, jumping up, out of the sleep, out of nowhere. And we could quantify and could find, okay, how many events do we see? And we clearly can show that the number of events is depending on the genetic background, because we previously found that when in the classical way of breeding, they are on a like 6 background, they don't show the severity as we see on the 129 SV background or other conditional knockouts. And that is indicated here. Yeah, so we have twitches and jumps, and the frequency is the highest in the conditional one, but it's also higher in the SV background. So this plays a major role. So what we see in these animals, and I will show you later how we do this, we again place recording electrodes on the motor cortex and on the cerebellum, and we can now measure nicely slow wave sleep, we can measure awake states, but we also observe spike wave discharge. Actually, this is unique because we only observe this in the animals. And we observe this in a way where it is not always coupled with twitches. So it's a low amplitude signal, but it can be easily picked up, and we know it coincides to a large degree, but not necessarily. And this is a very nice, fantastic approach where you now can remotely, you can program, you can extract, you can use templates and to quantify, and you can see how the spike discharge wave in a so-called ECG, or we should be better saying electrocardiogram signal, is available and tells us that this can be measured. Actually, we use now also drug treatments with anti-epileptics that are also used in these children, and they are rectifying the situation also and improving the situation for the mice. So this is a very nice approach and very useful because you get fantastic data because you're doing this in the home cage. The same applies for white matter disorder models. So you here see the motor differences in clasping because these show also differences, and this clasping behavior is more or less absent in the wild types. It occurs in the MLC1 knockouts, in the GliaChem knockouts. Here, a schematization. This is the skull surface. We insert a screw. This is sitting with its end on the top of the dura above the cortex. This is coupled to the transmitter. This is de-insulated by a UV hardened dental cement. And you can nicely look at the progression, and you can do a lot of studies longitudinally. We have mice running around with these transmitters for 3 months. And since the coat is covered on top, there is no inflammation. It is really working fantastically. So in the MLC mice, we could nicely see, of course, these discharge patterns as shown here. This is now a relatively long trace. This is zoomed up for a little segment, and in the segment, you can see this discharge pattern. And the same applies for the GliaChem mice. And if you look at these interictal spikes as we refer to, of course, we can see that they are absent, and these are present in both mutants to a high degree. Of course, there are individuals that have a low level as well. Now, the issue is, in these animals, we did not see very much seizure-related aspects, so we decided we would challenge the system in order to find out what is happening if we stimulate with kinates to bias the signaling into a more excitatory domain. And as you can see now, with increasing time, the racine score increase, which is an index of the severity of seizure-like occurrences, from very simple mouth movements, which are described at least in humans, which are delicate in mice, up to higher levels. And this is the range. And when we did this, what we found out is the mice in the wild types increased to a level to 2 roughly, and then returned, and the others progressed and had to be in the end terminated because it was not ethically acceptable beyond that point anymore. And that's nicely shown here in the spectral analysis, where you can see after kainate injection, there is some change, but it will return to baseline, whereas in the MLC, you see some change, and then you see a massive increase, particularly in simple and complex and intermediate spike frequencies with increasing time, with this increasing power, as you can see. And this is shown here with the quantification, where you can nicely see this is a very useful approach for us to quantify and to now look at therapeutic interventions to see whether these challenging conditions can be rectified or abolished under certain conditions. Of course, these are global mutants now, different ones are used, but this is a very useful approach where we can now use these animals. And in this case, we know now that the swelling may be related, and this may be related to a change in the potassium concentration in the brain, which is associated with, of course, a buildup of water, loss of white matter. And this is now electrophysiologically investigated in more detail to find out what the actual mechanisms are, because at this moment we are looking at the symptoms, we are looking at the consequences, and we are now trying to understand what are the mechanisms to induce this in the first place, to have a much more targeted pharmacotherapy to interact with this early on. And this brings us to the issue that we tested the monk 18 mice also on the dynamics, but we did not find any crosstalk between the epileptic seizures and the autonomic system. But I want to draw your attention to this nice paper from 2008, where, of course, people measured on the top the recordings of the EEG, and they also looked at certain discharges, and they looked at the ECG pattern in synchrony. And of course, we know that there are differences where cardiac arrest is linked to, of course, dysfunction of certain channels, such as the calcium or potassium delayed rectifiers. And of course, that means that these patients then need pacemakers or defibrillators, because of course, if you have these long gaps of hypoperfusion, does the system restart? Does it come back or not? And this is of course related to the so-called sudden unexplained death or SUDEP. Huh? And this is of course the case where in another study, this channel, and this is the paper that I referred to before, is highly linked, because you can nicely see that there are particular discharges in the cortical region, yeah, where you have a mutation, and you see that this results in a conduction block. And this tells us that, of course, and this is the major issue, it's not by chance that many, of course, psychiatric diseases have a linkage between epileptic activity, anxiety, and cardiovascular problems, because the network, as I will show you, is of course highly conserved, only that we have focused in the behavioral domain on the forebrain areas. Actually, we know very little about the contributing factors on the basal function. If we see basal functions will affect this, we will of course easily be able to diagnose this also on the autonomic output system. There may be a dissociation, because it is known that under strong emotional challenges, the system can be driven in a wrong direction. There are fantastic studies that show that you can have in pigs with a malfunctioning heart that is brought under stress, you induce arrhythmias, and these arrhythmias of the heart can be eliminated if you deactivate transiently the amygdala. So that means that there is a connectivity between the prefrontal and the amygdala areas to the output areas of the autonomic nervous system that contribute to dysregulation, where it's very important to acknowledge that we are dealing with one system that only has different output functions, and as usual, we should be aware that we should combine different biomarkers that classify then disease models. And therefore, I think it's important to note that, of course, the ECOG signals provide useful information on seizure and epileptic-like activity in general models. They show higher sensitivity to detect seizure-like neural activity in the absence of clear behavioral symptoms, which is very important. They can be complemented with pharmacological challenges to determine the seizure susceptibility, because we know in many disease models, under non-stressed conditions, the system seems to work well, but under challenging conditions, the things go wrong. And of course, the EEG measurements and also the heart rate measurements are nice, because you can look over longer time periods relatively remotely to look at changes over time, particularly when it comes to interference with pharmacological interventions. And I think that this combination of this is really important to look up in the future. Again, I have to thank a lot of people that helped me with this. And this is, of course, as usual, a big collaboration. Of course, we are still having ongoing research with psychiatry units, because it's more the psychiatry unit that interestingly is interested in these aspects, rather than psychologists or neuroscientists. For them, it appears to be a little bit too complex, it seems. But I think what I want to show you is these measures only work if you have an intact animal. You need animal research for this, but you have to make sure that the animal conditions are in a level where you are in a physiological realm. You cannot use restrained animals for this with pharmacology. You need normal animals for this. And with this, I'm finished. And I again thank you for your attention, and I'm looking forward to your questions.
Thank you very much, Professor Oliver. Now session is open for queries.
Sir, I have one question.
Yeah.
What is the normal mean heart rate in case of mice? As we can see from your some of the research papers that, for example, in the selective serotonin receptor inhibitor studies, that you have shown the mean heart rate from 300 to 600 BPM.
Yeah, I can tell you it strongly depends on your housing conditions. If you have single-housed animals, their baseline would be roughly in the range of 500 to 600. If you have group-housed animals, during sleep, the heart rate goes down because they are better, let's say, have a better thermal insulation. And you have to really watch out because at some point you come into the animal facility and you have a very low baseline, and then you find out your temperature regulation is low, because you may know that the thermoneutral zone of mice is roughly 27 degrees or 29 degrees even. And the higher the temperature in your animal facility, the lower the baseline heart rate of the mice. So you have to know, yeah.
Yeah, is it that the heart rate in case of rodents is much higher as compared to human beings? Generally, in human beings, we have a range around, it varies from 60 to 100. And in mice, you are showing 500 to 600, means almost 6 to 7 times more.
It's 8 times more. The maximum heart rate in the mice is that we have measured is about 800. And in many
Don't you
studies,
think it's very high?
It's very high
as compared to human beings.
Exactly. It's very high. But of course, be aware there is this negative allometry between body size, metabolism, and heart rate, because you have small miniature shrews, or even just think about hummingbirds. I think their heart rate goes beyond 1,000 beats per minute. It's a message.
My second question is how far heartbeat is a reliable parameter? For example, if we take example of human beings, generally we see there is a variation of even normal heartbeat. It varies from 60 to 100. So there is a variation of almost 50%.
Yeah.
So how far it can be considered a reliable parameter?
This is the reason why I said I would go away from the means, because the means are meaningless. Yeah? It is the matter of the dynamics. Is it a healthy dynamics? And the problem already emerges because there are many human studies that show significant effects with a heart rate change of 5 beats per minute. While knowing what I showed you, 5 beats per minute is in the normal range of the fluctuation. I have done classes with students, and I would argue that many of the studies show significant effects, but physiologically meaningless information, because it does not really care. 5 beats per minute for a human being is nothing. It is not relevant. Yeah?
My another question is, other than heart rate, did you check the pattern of these ECG peaks? As a layman in our school days, we have studied PQRST peaks and all.
Yeah.
So, and is it similar to human beings in case of rodents?
It is similar to humans. It depends, of course, on the position of your electrodes. Yeah, because we use normally a type 2 recording, and this is more or less identical. Of course, there is a difference in the T wave, in that the T wave is faster. And therefore, you have to be aware. What I have not seen so far is ventricular tachyarrhythmias. We see mainly ventricular bradyarrhythmias at a rate of 1 per 10,000. So I have quantified this. And this is, of course, therefore, of course, you have to be aware. Therefore, mice are not a good model for tachyarrhythmias, because the heart rate is so high that the heart re-excitation, re-entry is not as dangerous as it is in humans. Yeah? And therefore, the risk of ventricular fibrillation is much lower.
Okay. Often I wonder one thing, despite the importance of EEG or ECG, ECG recording, normally it is not much in clinical practice, at least in India. We have seen that the doctors still rely on biochemical parameters to diagnose any particular disease, rather than relying on this ECG and EEG reading. They go for ECG only when they suspect there is some cardiac abnormality, but not for epilepsy or some types of such other types of diseases.
Yeah.
So what is the cause for that reason? Is it not very reliable?
I think the problem is that, as usual, most people want to have a short measure and they want to have a brief result in no time. My dilemma in my measures is, for example, if you get a short recording, it does not tell you anything. You need long-term recordings to get a real good analysis on this. And I can tell you, I have done analysis from ECG Holter recording systems over several days. And I can rely on the software that is on the systems, or I can process the information. And you need to have a control on the raw data. And of course, the clinicians do not look at raw data. And there are a lot of bugs. And the bugs don't matter for the means, but they matter for my nonlinear readouts. I can give you, I have given a course on this as well. And I have even instructed in the meantime cardiologists to take care about this. And we have shared this for that reason. Because believe it or not, I have analyzed data where I received data from an ECG holter where I saw ventricular tachyarrhythmias, where I had to tell the clinicians, send this guy to the intensive care unit because he's at risk of ventricular fibrillation, because the systems don't detect it. The devil's in the detail. Yeah. So what we want is a system where you record this, you get your clean answer, but that does not exist, as you know. We have to be careful. And therefore, the more complex it becomes, the bigger is the inhibition of people to follow this path. This is the problem. And unfortunately, we are dealing with complexity, and we are trying to understand complexity with very simple tools that, in my view, are often insufficient.
In fact, but this could have been a much better technique, because this is not an invasive technique. Rest of the whenever you go for biochemical parameters, you have to inject, you have to exercise something, you have to perform surgery. But this is a non-invasive technique, and I think it's more reliable.
Look, I have toured in Switzerland the pharmaceutical industry, and they tell me, since it's not legally required, why should we use it? Because they are afraid that this measure will pick something up, as I showed you for the SSRIs. That is something that they don't want because it will hamper their selling of products. Yeah?
Or is it due to lack of understanding that most of the physicians don't understand these waves and peaks? Because it's very tough. You get peaks like that and then to interpret something from that until there's a very expert in it.
I can tell you this is the reason why we have engaged always in collaborations. If people, and I have many collaborations where we have used an septic shock model where, based on these nonlinear measures, you can predict the survival of the animals before the animals are even dead. Yeah. So I told them we should use it more in the clinical setting. The problem is that there is not the technique available, nor are the resources available, nor is the time available. And this is at this moment the biggest problem. And I have worked with this also with people that undergo such a complex cocktail of pharmaco interventions that the baseline is already screwed up before we do measurements. Yeah, Parkinson patients, for example. It is not easy to really get to the bottom of these things.
My question is, how easy is it to perform all these experiments, record these experiments with rodent system? Because when you perform an ECG with a human being, you generally ask the patient, okay, you lie down, patient lies down, you put all the sensors and all. But I mean, working with animal system, is it very easy?
If you
want, and all.
Yes, if you want to measure the ECG and the EEG or the ECOG in mice, it's about where to implant the transmitter. The new transmitters, if you have relatively large mice, it's really straightforward and relatively easy. You will manage. I would invite you, and in 3 days you can do it yourself, for sure. Okay. So if any one of you needs a SOP of how things are done, check with me, because I can provide you with documents, and I've published work on this, and we have internal standard operating procedures, what we advise to do, which cement to use, and so forth, because there are a lot of technical details that make the difference.
For example, nowadays we are having portable ECG machines. So is the ECG machines used in case of these rodents, they are same that is used in case of human beings, or there are some difference, or there is some custom made?
Actually, there is a company which is called Data Sciences that I work with for quite some time, and I don't want to make advertisements for them because I'm completely independent. I have used their transmitters because they are really the best. And I use customized systems to analyze it from other companies. So it's a patchwork, and in some points, there is a fantastic website called physionet.org, where you can download the analysis modules, the software packages, the instructions. It is fantastic. It's out there. You can educate yourself, and if you are smart enough, it is fantastic. Now comes the point. Most people that can do that get a job at a stock market business, and they get a salary that is 5 times as high as a scientist. And therefore, the people that really do this are too poorly paid, and that's the reason why we are lagging behind in science. Actually, that's the truth.
So my question was, the instrument is same in case for the one that you have used for mice and the one that you use for human beings.
Yeah,
It's same.
it's the same. It's exactly the same, because the system does not care what the intervals are, because it cares about the dynamics. And the dynamics is what I told you, and what I want to have you learned is what is fully translational. The dynamics of mouse and man is the same, but the heart rate is not the same. So if we now would do interferences with pharmacology, we should say the same effect, and we can draw conclusions. But what we have to find out is now we have to do more brain-specific temporal interventions, and then look at the cardiac dynamics to see what actually is the cause, because we have a nice diagnostic tool, but we don't know yet what to do. Huh?
And sensor is also same. And number 2, the rodent systems have got a thick layer of hair on their body. Doesn't it interfere with the signaling and all?
No. Of course, you have to watch out. If you have the telemetry system, you have to watch out that you have no disturbance signals in the neighborhood, and you should watch out from metallic devices. But the rest is relatively straightforward. Yeah? So I have recorded this, and I have trained many people on this, and I have trained many people how to use it. You are welcome. If you have further questions, contact me, and I can send you information, because at some point I have a hard time, because there is a problem in the sound transmission that is not optimal. But
thank you, sir.
Don't hesitate, and don't be too formal about this. If you want something, send me an email, and I will give you updates on this.
Sure, yeah? Sure, sir. Thank you, sir. Thanks a lot.
Any more questions? What was that?
Yeah.
So with your kind permission, can I ask one last question?
Yeah, you can ask.
Okay.
Yeah.
So it was a brilliant work which you have summarized in a very short time, and I congratulate you for that. And you have shared a lot many things that we need to know and learn in animal behavior, definitely. And just a query about that is a personal query, that my father is suffering from schizophrenia, and often his heart rate is correlated sometimes with the onset of the disease. So why is this so, that in schizophrenic patients we often see the altered heart rate? My topic is quite different from the story, because it's my personal question. I just wanted to.
Yeah. So let's be honest. The heart rate is not an index per se for PTSD or whatever. We have to be very careful. And I would go away and say what we have to develop in the future is diagnostics and characterization of so-called biomarkers in clusters of biomarkers. Yeah? A biomarker, if I would send you a paper, age is a factor for lack of neuroautonomic control. The older you get, the worse the connection. People that are alcohol addicts have a decline in the neuroautonomic control. There are many diseases that play a role in this. And there are sometimes easy assays to check how this works. For example, orthostatic blood pressure regulation. So what you measure is the heart rate when you are standing, and then you lie down, and then in this transition phase, you see the feedback of the brain. And this is not very well understood. And I think this would need a lot of better understanding, because as you see, it's the brain-heart interaction that matters, and we have not understood how the network in the brain really works. We have some tracing studies. We know the overlap with the behavior, but there are very few approaches that have really addressed this in a decent manner. And schizophrenia is very delicate anyway to begin with, because we are look at the real animal models on this. They have some rudimentary effects, but they are not really good, in my view.
So my second question, just a bit query about to start with these kinds of experiments like ECG or EEG on mice. What acclimatization period should be given just after the transference of animal from another university or a place where you are residing? So acclimatization time would be differing from like 2 weeks, or usually we gave it 2 weeks for acclimatization.
If you're giving 2 weeks, then you are fine. It matters, of course, depending on the circadian change and whether they have been completely in a box for some time. And then I would say 2 weeks is fine. Of course, that's the disadvantage of some of the surgeries. If we do now invasive surgery, we also recover the animals, let's say minimally a week, normally up to 2 weeks before we even get going. And there are a lot of papers out there where, for example, there is a paper, a highly cited paper out that reports that the baseline heart rate of mice is 700. And if you use a beta and parasympathetic blocker, nothing happens. Yeah, if the heart rate is 700, this is not normal heart rate. It's the animal was in pain and was still activated. And then if you use a parasympathetic blocker, nothing will happen because there is no parasympathetic tone. So you have to watch out what you do. And the baseline of your heart rate tells you already something about where you are. So you have to be very careful. Yeah. And as I told you before, it can be as simple that you are having a very high temperature in your animal facility because your temperature regulation system has failed, and you are having today 24 instead of 21. Yeah. The animals are grateful for this because they drop their baseline metabolism and therefore the heart rate goes down substantially. You will be amazed. And this is why the mice are always very sensitive, and you have to watch out when you do these experiments. I have created rooms where we have a tower with all the electronics, and then we have a funnel to the next room where all the experimental animals are sitting there. We are not going in. We only go in if we have to really interfere, and we otherwise do not disturb the animals anymore.
Thank you so much, sir.
Yeah, you're welcome.
Sir, actually, I have one question.
Yeah, go ahead.
Sir, it was a very informative lecture. Actually, my question is, some of the reports suggest that most of the heart attacks occurs during the sleeping condition. What is the reason behind that, and how brain cells regulate heart attacks?
First of all, I do not quite agree. In my view, the highest risk of heart attacks is in the early morning when the high vagal tone is coinciding with an increase of the sympathetic tone from your early activity. So shoveling snow in the winter in people with obesity is not a good suggestion. Yeah? The second thing is, we know that particularly heart occurrences occur after emotionally challenging issues. Just look at earthquakes, unusual sports events and things like that, and you will see that thereafter is a peak. So that means the autonomic or the emotional system plays a role on the output. And I mentioned to you before, it's an older paper. If you have a maladaptive of a malfunctioning heart and you get an hyperexcitation via emotional stressors, it can lead to arrhythmias. You all know tachycardia, but it can lead to arrhythmia. And in pigs, it was shown if you deactivate the amygdala, these arrhythmias are gone. Yeah? So the whole network that we talked about first in the fear learning is really coming back in the cognitive emotional regulation on the autonomic system as well. And whether it is the areas that are activated by epilepsy that drive this, we don't know yet, but it's very likely. And there you can combine this in one go.
Yes, sir. Thank you so much, sir.
I think all questions are been answered. Now we are moving to next step. So now I welcome Dr. Rajneesh to summarize both the sessions.
Good afternoon to everybody. As we've already running short of time and we are into the time slot of the validatory functions, I'll keep my summary short and brief. And so this was a very informative session, and we looked at animal behaviors, even in uh, animal behavior changes due to neuropathy caused by diabetes, and astaxanthin, and what are the effects of that astaxanthin on behavior, and as well as the biochemical parameters was briefed by was talked upon by Dr. Surabhi Bajpai, and that was a very good session, and it gave us the information about astaxanthin and the health benefits of astaxanthin with respect to neuropathy. Thank you very much. And Professor Oliver has given a very comprehensive and detailed idea about how animal behavior experiments should be planned, designed, and what are the things that we need to take care of. And it included from the time points to the handling, and also the baselines of your parameters that you are measuring. And the very interesting aspect which I thought was the relationship between the brain and the heart axis, and his implant which measures the ECG was very nice. And you can see that if you develop a model, or if you develop a system, how great data that you can generate. That was shown by him. And he also talked about what are the various home cage systems and how to refine those home cage systems, and what are the parameters that affect these home cage systems. While studying animal behavior, he also talked about linear versus non-linear comparisons of your parameters, and what are the advantages and disadvantages. I am not an expert in this area, but it was very intriguing and interesting area. I'm sure that all the participants have gained a lot of knowledge, and have been benefited by his excellent talk, and very informative and very comprehensive. Thank you very much. With that summary, I hand it over to the chair to conclude the session. Thank you.
Thank you very much. Now I invite, actually on the behalf of this committee, I again thankful to Professor Oliver and Dr. Surabhi Bajpai, and the session was very well conducted, and I think we are not too much aware about this time.
4 o'clock.
So I think now we can go for valedictory session, and if Professor Janakahi ma'am want to say something.
And I want to thank Oliver. Thank you so much, Oliver, for accepting our invitation and being with us. It was really great. Thank you so much.
It was a pleasure to be here, and thank you for the invitation.
Yeah.
And as I said, feel free to distribute my email, because science is about sharing ideas and sharing concepts, and stimulating others to be inspired to follow some of these lines. And it's as usual, it's always good to look over the fence. That's my strategy. Yeah.
Thank you so much. Thank you.
Thank you, sir.
Thank you very much.
Rajkumar, I think valedictory session you can start.
Divya will conduct.
Okay.
Thank you, sir. Thank you, Oliver, sir, for the nice lecture. I would like to invite Dr. Rajkumar Koiri for the valedictory session, the organizing secretary of this webinar. Please, sir.
My warm greetings to all of you on the occasion of validity session of this international webinar on neurobiology of behavior, the challenge and the promises for translational research. Dear participants who participated from all over the country, all the respected speakers, both national and as well as international, my seniors, colleagues, and friends, and all those who have joined us on the virtual platform. On behalf of the Department of Zoology, Dr. Hari Singh Gaur Vishwavidyalaya Sagar, and on my behalf and on behalf of organizing team, it's a great honor and privilege to propose a vote of thanks on this memorable occasion. Let me first of all start by giving a glory to the Almighty God for making this event a resounding success. First and foremost, I thank our Honorable Vice Chancellor, Professor Nilima Gupta Madam, who, despite her busy schedule, found time to grace this occasion with her blessings. I extend my hearty thanks to all the esteemed speakers who accepted our invitation to grace this 2 days event with their precious knowledge and making this webinar a great success. We extend our heartfelt thanks to Professor Elena Rodlevic Madam, our keynote speaker, College of Medicine, Rose F. Kennedy Center, Bronx, New York, USA, for enlightening us with her talk on brain, mind, and behavioral models of psychological processes. In her 2-hour enlightening deliberation, she talked about basic organization of the brain, different models to study psychological process, problems and challenges related to the use of animal models, and the most commonly used paradigms to study psychological process. How can we forget some of the highlights of her talk? Brain as an electrical network, but much more beautiful and regional nodal network. Best part was when Professor Janak Dulariahi Madam requested her for a talk. She happily proposed herself that I will give a talk of at least 2 hours. That itself speaks a lot about her personality. Okay. Sorry. इसके पहले? Next, we have with us respected Professor Sukla Prasad, sir, from Banaras Hindu University, who also happens to be my teacher par excellence above. In his enlightening lecture, sir beautifully explained all the associated pathways before raising some key questions associated with hypoxia-induced spatial memory impairments and associated alteration in AMPA, glutamate receptors, trafficking proteins like glutar 1/5, neuronal cell density, dendritic spine density, and how they can be reversed with Bacopa moneri extract. This was followed by oral presentation session, and keeping in mind the time constants, we tried to accommodate as much oral talk as much as possible. There were 12 excellent oral presentations, mostly by research scholars, with beautiful histology, same images, enzyme assays, synthesis of neuroparticles, neurobehavior, etc. The oral presentations were judged by independent experts. According to all the judges, all the presentations were very good. And it was very difficult to differentiate and categorize them as first, second, or third. So they have selected 4 best oral presentation without any distinction, whether it's a first, second, or third. So they selected 4 best oral presentation, and the name are as follows: Swati Jain, Roshni Rajput, Tanmaya Rani, and Devrata Das. My heartiest congratulations to the speakers who have been selected for best oral presentation. They will shortly be provided with the best oral presentation e-certificates in a few days' time. Here I would also like to mention that soon we'll share the feedback link to all the participants on the WhatsApp group and also on their email ID in a day or 2. After you fill the link, or the Google feedback form, you will receive e-certificates on your email ID. So the filling of feedback form is mandatory for receiving the participation certificates by all the participants. Next, we have our session 3, in which the first talk was delivered by our eminent speaker, Dr. Aniruddha Mitra from Sarojini Naidu College for Women, Kolkata, India, who delivered his talk on the test of smell. Drosophila detects orders through test neurons of proboscis. He is such a humble person that without any ifs and buts, he accepted our invitation of talk in a short period of just 3 to 4 days before the event. We tried to make the program as much diverse as possible, staying within the ambit of the theme of the webinar. And this was achieved with Dr. Aniruddha's talk. His talk was so impressive that as soon as his talk was completed, I started receiving call from students and participants that, sir, please request Dr. Aniruddha Mitra, sir, to please share his presentation with the audience. I would request Dr. Aniruddha Mitra, if he can, then please share his presentation to all these participants. This was followed by invited lecture from Dr. Akash Gautam from Center for Neural and Cognitive Sciences, University of Hyderabad. In his talk, Dr. Gautam very vividly talked about the role of immediate early genes in regulating the learning and memory processes. His talks was especially helpful to all the upcoming students of neuroscience. And before his talk, we have requested him to keep his talk as simple as possible so that the student community is benefited. And he amply did it. As Dr. Yukesh Bhargava rightly said yesterday, that seeing is believing. And in the following talk, Dr. Santosh Singh from Guru Ghasidas University very beautifully narrated neurochemistry of hepatic encephalopathy with beautiful videos and images. This was followed by the talk of young dynamic upcoming neuroscientist Dr. Kiran Pandey from Albany Lab, Center for Neuroscience, New York University, who talked about the complex topic, role of autophagy in memory formation, but with simplicity par excellence. Today we started the first session with Professor Meena Kumari from Kansas State University, Manhattan. Madam in an enlightening lecture highlighted the various multifunctional proteins and their impact in neuronal physiology and addiction. She discussed various aspects of protein folding of nascent glycoproteins in follicular development and oocyte maturation. This was followed by the talk of another young dynamic onconeuroscientist, Dr. Devra Dixit from Sanford Berhan Device Medical Discovery Institute, California, in which he very beautifully explained epitranscriptomics and how it serves as a new RNA code for the regulation of gene expression in an animal model of glioblastoma. Dr. Dixit discussed at length about the new evolving research area of RNA modification and profiling of glioblastoma stem cells and neuronal stem cells, particularly RNA M6A, METIL, YTH, DF. In the last talk of today's first session, we have with us Dr. Prashant Modi from Enopoya Research Center, Mangalore, who very beautifully illustrated the application of multi-omics technology in deciphering brain function and diseases. He discussed extensively about quantitative proteomics, brain proteome map, and molecular targets for traditional Ayurvedic medicines. In today's last session, we have two lectures. The first lecture by Dr. Surabhi Bajpai from Banasthali Vidyapith, in which she discussed how astaxanthin reduces oxidative stress and alleviates diabetic neuropathy in STG-induced diabetic mice model. And in her presentation, she beautifully illustrated with suitable examples of the different neurobehavioral experiments and with beautiful videos. This was followed by the last invited talk by our guest of honor, Professor Oliver Stiedl, sir, from VU University, Amsterdam, Netherlands. His lecture was divided into two parts. In the first part of his lecture, he introduced us to the classical behavior test to assess emotional states of mice from anxiety to fear based on associative learning and its extinction. In the second part of his lecture, he talked about various examples of studies based on autonomic, that is ECG and electrocardiogram measurements based on radiotelemetry. He discussed behavioral, autonomic and neurophysiological phenotyping of animal models from health to neuropsychiatric diseases. His study on ECG radiotelemetry in freely moving mice was really interesting. Thank you, sir, for enlightening us with your vivid, detailed lecture. On behalf of Department of Zoology and the entire management group here, I extend a hearty gratitude to all the speakers for sharing their graceful thoughts and knowledge with us in this webinar. I extend my gratitude to our convener of this session, Professor Janak Dulari Ahi Madam, who was behind everything associated with this 2-day webinar, right from its inception to its successful execution. Thank you, ma'am. I also extend a heartfelt vote of thanks to our young, dynamic co-organizing secretary, Dr. Divya Rawat, for this event, who took all the pain from day one, right from creating Google Forms to designing certificates and communicating with the audience. In fact, she will be the one who will be engaged with the webinar till the certificates are dispatched to all the participants. Thank you, Dr. Divya Rawat.
Thank you, sir.
I must thank the organizing team, volunteers for working hard for the past few days to make this webinar successful. No program can be successful until and unless you get the support of your young, enthusiastic student team. I am thankful to all the research scholars of the Department of Zoology, particularly Rais Ahmad Naik, who, despite being in thesis submission phase, took out all the pains in making this event successful. Nidhi Gupta, Tanmaya Rani, Lekhan Lodhi, Debrat Das, Rosni Rajpur, and Binondi Dubey for all their logistic support and media briefing. I also extend a hearty thanks to all my senior faculty members, Professor Subodh sir, Professor Varsha Sharma madam, Professor Sweta Yadav madam, Dr. Malvika Sikdar madam, Dr. Dipali Jat madam, Dr. Payal Mohbia, and Dr. Rasmi Srivastava madam, and Dr. Yogesh Bhargava from Microbiology Department, Dr. Aswani Kumar from Department of Botany, and Dr. Rajnish Anupam from Department of Biotechnology for successfully conducting all the sessions as the chairperson and rapporteur, and for their constant support as and when required. So thank you all of you. I also extend a heartfelt thanks to Professor Subodh Jain, Head Department of Zoology, and Professor P.K. Khare, Dean, School of Biological Science, for providing immense support to make this webinar successful. I'm also thankful to all the non-teaching staff members of Department of Zoology for their constant support. At last, I congratulate all the participants who came from different parts of India and for their active participation and making this event successful. We have more than 220 registrations. So thank you everyone once again for making it a great success. Hope all of you enjoyed all the deliberations. In the process, if I have missed someone's name unintentionally, I extend a heartfelt apology on my behalf. Thank you everyone, and hope to see you again in near future. Thanks and warm regards on my behalf and on behalf of Department of Zoology, Dr. Hari Singh Gaur Viswanath Sagar. So thank you everyone.
I would like to say that Dr. Rajkumar has thanked everyone. So I would like to thank Dr. Rajkumar for being present and for carrying out the webinar very successfully. Dr. Rajkumar, thank you so much.
Thank you, madam. Now I hand over it to Dr. Divya Rawat.
Thank you, sir. Thank you, ma'am. I would like to all stand up for the national anthem. Please stand up for the national anthem.
खुद ही गा लो ना, खुद ही गा लेते हैं. चलो चलो चालू करो. भारत भाग्य विधाता, पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल हिमाचल यमुना गंगा उच्चल जलधि तरंगा सेशन एंडेड. हाँ, अब मैं Okay, thank you everyone. I thank all of you, and the webinar is closed now. So hope to see you all again in some other platform. Goodbye to all of you.